Good morning and welcome again to our day 3 and the final day of our CRB Science and Management Symposium for 2025, everyone. Okay, just in terms of our partners organizing the CRB Symposium, these are the organizers' logos for your information. Okay, just highlighting again the planning committee members. We have Rita Gisau, Franny Brewer, myself, Dr. Keith Weiser, Dr. Elliot Parsons, and Dr. Mike Melzer. Okay, just in terms of our program, if you have not downloaded that, that's been shared. So you can also use the QR code to download the program, but it's been posted, shared as well. Thank you. In terms of the day 3 programs, we have the welcome back and opening talks, and then we have a break. And after the break, we have session 3, which is continuation from day 1, and that will cover detection and monitoring. We have 6 talks for that, which will have 12 minutes each to share their talks. And after that, we have a break, and we have session 4, which will be on insights from the Pacific. And we have a total of 3 presenters for that. 2 presenters will have 12 minutes each to talk, and we have a presenter that will have 1 minute talk, and then we'll have the closing remarks and finish our symposium. Thank you. Just before we start, in terms of the ground rules, again, just to remind us, add all your questions into the Q&A box. Yeah, so we can have them concentrated there for us to answer. It gets a bit, or if you send it through the chat box, that's only coming to us, the moderator. So we may have issues. So just make sure you add your questions in the Q&A box. Okay. Otherwise, with that, we'll get started now. And I now have the pleasure of introducing our first opening session speaker, which is Mr. Alan Aku. Mr. Aku is the Managing Director for Papua New Guinea Kokonas Indastri Koporesen, KIC for short, which is the coconut authority of the country, and is also the chairman of Pasifika NiuNet, which is the Pacific Regional Coconut Governance Body. And it will be presenting this morning in his capacity as the chair for the Pasifika NiuNet. And over to you, Mr. Aku, to proceed with your presentation. Thank you.
Thank you, Mark. Let me put my slides on, and then we'll start. Okay, am I on all slides? Can you see my slides?
Yes, you are.
Okay, thank you. Thank you. Firstly, let me thank all the organizers and coordinators of the symposium for the coconut rhinoceros beetle. I'm glad that we can discuss this pest because it's a problem across all of the Pacific. And so it is good that we share ideas in this area. Let me thank all of you for giving me a time slot to do a presentation on the Pasifika NiuNet, which is a mechanism to support our local coconut sector in each of the Pacific nations. The Pacific NiuNet, Pasifika NiuNet, was recognized by the Pacific Heads of Agriculture and Forestry Services in 2017. They saw the importance of coconut across the Pacific, not just for commercial, but also for food security. And I think most small island countries use coconut as food more than just exporting it as an agricultural commodity. And so its importance goes down to every community in the region. And so the Pacific Heads of Agriculture and Forestry Services realized the importance of this crop and need for development work in the region, especially on the research front. And so the Pacific Community, or SPC, was directed by the hosts to conduct consultations with the coconut experts across the member countries, and just to understand the coconut sectoral ecosystem. And in that consultation, it was realized that there should be a platform, high-level platform to guide the strategy, and also for resourcing of the R&D. And during these consultations, it was identified or realized that it is important for this platform to be set so all the countries can be able to have a point of contact where we can share ideas and also share expertise. And so the consultation took place in Fiji, and the Pasifika NiuNet platform was born in that place. So that's basically how the Pacific NiuNet started. The Pacifika NiuNet structure is we got 12 representatives on the Pacifika NiuNet, 6 private sector. This is two from each sub-region, which is Melanesia, Micronesia, and Polynesia. And so we have 6 private sector reps and 6 government reps on the team. We have a chair, which is currently myself, and we have deputy chair, which is Dr. Mark, what's his name? He's from Samoa.
Posui. Dr. Posui.
Yeah, Dr. Posui from Samoa is the current deputy chair. And we have 6 or 2 reps from each of the sub-regions, 1 private and 1 government. And then we also have leaders. We have pool of expertise in areas that have been identified. And these areas include genetic resources, sustainable forestry and landscapes, sustainable agriculture, markets and trade, and policy and governance. There are a pool of experts in this area, in the pool. There is one leader of each of these areas appointed, and under him there are 5 members which have been identified with their expertise in these areas. And so experts are put into these 5 groups according to their qualifications and their expertise. What our values and guiding principles in the Pasifika NiuNet are on the screen, especially on the area of sharing. We'd like to share with one another, not only in knowledge, but in expertise. And should there be any need from each of these countries or member countries, we can pull our expertise together to share ideas and even to go down to each of these countries to do surveys or share ideas in that country and try and find some solutions to areas that are of concern. So the guiding principles are there: sharing, we respect one another because we're from different countries. We respect one another. We respect in terms of the laws of that country, or we respect one another in terms of our expertise. We also see trust. We must trust each other, and we must be united in diversity, because we have different cultures, different ways of lifestyle, beliefs, and so we, in the midst of diversity that we must have unity, and we must deal with each other honestly and transparently. So those are the guiding principles with all the other smaller areas that of concern. What we have achieved so far in the Pasifika NiuNet, the establishment or the governance structure has been set. I've shared that one already in my earlier slide, that the Pasifika NiuNet governance structure is in place. We have developed a strategic framework to guide us over the next 5 years. And there was also a conference that was co-hosted by the Pacific Community and Solomon Island National University. This brought together all the coconut experts to share experiences on research and management of coconut pests in the Pacific region. I think this was triggered mainly because of the coconut Rhinoceros beetle that was devastating many coconut industries in Pacific countries. And so this symposium was called, but during that conference, they also looked at other pests that are in the region. The conference informed, as discussed, and then they developed the action plan for prevention, containment, and management of priority pest and disease in the region. And so this was tabled after the conference at Solomon Islands, tabled at the governance board, and it was endorsed. So that has become part of our work plan in the next 5 years. Our next steps, we're working through the Pasifika NiuNet proposals to mobilize resources. We already have an action plan, but that needs to be implemented. But the implementation of that action plan needs resources. So we are doing that at the moment. We are drafting proposals through our secretariat based at the Pacific Community, pulling together proposals to mobilize resources to support our action plan. The top 3 focuses in the area of our work in the region is firstly, we must do the limiting survey to see what kind of pest and disease are present in the region and the status of the pest presence in each of the countries. So a delimiting survey needs to be done. And we also must, after the delimiting survey, there must be biosecurity plans and emergency response plans that must be put in place. In PNG, we already have a biosecurity plan, which is shared with the Pacific community and to be passed to all the member countries. So we have a biosecurity plan for coconut. And we also have emergency response plans. So we are sharing those ideas with our Pacific neighbors and through the Pacific community as the Secretariat of the Pasifika NiuNet. After that, we have to do biosecurity plans for each of the member countries and also emergency response plans. Our experience in PNG is that when Bogia coconut syndrome arrived, we didn't know how to handle the outbreak of Bogia coconut syndrome. And because we didn't have biosecurity plans and we didn't have an emergency response plans. And so that taught us lessons to develop this. And so we are sharing with our Pacific member countries that we must prepare for anything that happens. And, you know, coconut rhinoceros beetle is running, devastating many of the coconut industries in the Pacific countries. And we need to develop these biosecurity plans are important. On the area of capacity building, we also want to build capacity on the ground that farmers must be able to identify at least at its infant stage, they must identify the pests or the disease that appears in their farm, and they must know how to report. And so we are also building capacity on farmer level for reporting systems on coconut pest or disease. The delimiting survey that I mentioned earlier will help us to help the farmers in building their capacity to identify pests and disease that may appear in their farms or in their areas or in the country. I think that's about it for the Pacific NiuNet. And thank you for giving me this opportunity to share with you. And we are ready to accommodate territories or countries in the Pacific that are not under Micronesia, Polynesia, or Melanesia. So if you are a country or a territory that hasn't joined, we also encourage you to join us. And so we can join hands together to share ideas, knowledge, to help our communities and our farmers who are growing coconut in their farms. Thank you very much. Malo.
Thank you very much, Alan. And I think, yeah, you had 30 minutes and you're well ahead of time. So we'll see if we have any specific questions for you.
Thank you.
Yeah, participants, any specific questions for Alan? There is a question there, but I think it's more related to yesterday's presentation. So I'll ask for any questions that are specific to Alan's presentation.
Excuse me, Ma, is there one that about the picture?
Yeah, okay. There's one that has just come in. Where was the picture of the destroyed trees from?
Oh, this one. On my last slide. I think Carmel or Ramona would be the best people to tell me where they got this photograph from.
Alan,
or
I can answer that. That's from Vanuatu. Okay, there's a question from Elizabeth. Can a country in Africa also join?
Oh,
That's a big one. Yeah.
That's something that's interesting. Maybe we can discuss that at the governance board and see if we can accommodate interest from other regions of the world. But I think there is a bigger platform for all coconut growing countries, which is with international coconut community and the cogent or coconut genetic resources. So I think that's the platform that all other regions in the country merge or share ideas and so I think for Africa, we can direct them towards international coconut community to inquire if they can be part of that community.
Okay, thank you, Alan. There is another question from Trevor Jackson. How can we best involve local farmers in the pest response?
Yeah, I think I've covered that in my third, the last slide on the next steps and the third point. That we need to build capacity at the farmers levels and train them on reporting systems for the coconut pests. So the Pasifika NiuNet is actually working on that at the moment on how we can help farmers report.
Okay, thank you, Alan.
I think we also in PNG we also developing the MIS system for reporting of pest and disease or any other queries by farmers, and so we might share that on the platform, on the governance board and platform, and see how other countries can pick that up. It's a good system, and the data that will be reported will be real-time data. And you click on one province, you go and type the farmer's name, and all his information appears, or he reports that on a mobile phone and it appears on the system. So we might share that at the governance level for other countries, our member countries, to adopt. Thank you.
Okay, thank you very much, Alan. I can't see any more questions in the Q&A box. So I think we can, sorry, is anyone saying something? Yeah, if there's no more questions, then I think we need to again also time to try and accommodate one or two presentations that missed out yesterday. So we'll proceed on to our next presenter, and I will hand that over to Keith for the introduction. Thank you.
Okay, yeah, thanks very much. So we have, for our next talk, we have Dr. Carmel Pilotti. Currently, she's the curator for SPC's Center for Pacific Crops and Trees. She was formerly the associate scientist for coconut genetic resources, working on 2 projects to capture the diversity of coconuts in 5 Pacific Island countries. She set up the Coconut Integrated Program with the SPC Land Resources Division, resulting in the formation of the Pasifika NiuNet, who just heard a talk about. And she's now part of the Pasifika NiuNet Secretariat team within the SPC Land Resources Division. Today, she'll be speaking on action plans for coconut pests. And take it away, Dr. Pilotti.
Thank you, Keith. I'm sorry, I wanted to put my video on, but I can't find the while I'm talking. But anyway, okay, I'll just start. So thank you very much to the organizers for allowing me to present or give you an update on where we are at with our action plan for coconut. I think some of you were at the Coconut Pest Conference in the Solomon Islands, where a lot of the information for this plan was gathered. So I thank you all for your contributions to that. And it's sort of still in a little bit of a draft form. We want to sort of really get the wording right for it. So we won't be sharing it immediately, but we do want to share it with those in the CRB community in the future. So I think I don't need to tell you how important coconuts are in the Pacific. There's an estimated around 80 to 85 million trees, and coconuts basically are consumed on a daily basis in many of the Pacific Island countries and territories. And so they're very important for food security as well as income for livelihoods. So basically, we are, through Pacifika NiuNet, looking at strengthening preparedness and response and management of CRB and other major pests through collaboration and innovation. So from the conference, the Coconut Pest Conference that was held in Solomon Islands, we came up with 5 major pests that are of importance to the region. So these included the coconut stick insect, the leaf miner, boggy coconut syndrome, of course, the vectors of which still need to be researched, and of course CRB. And we are also concerned with other pre-border threats looming close to our doorstep in terms of the black-headed caterpillar and some ringcoffer species as well. So these were the pests identified. It certainly doesn't preclude any other pests that our member countries might think are of more importance to them. But these were sort of what we are starting to try to base our work on. So in looking at how we can manage these pests, we see that there are a couple of problems with current management of pests in general. And so the first problem is that management of pests is usually non-existent in many of our countries. Usually where low-level damage is observed, it's not managed at all. It's usually reactionary, so there's no real preparedness towards or anticipation of the threats. It's also project-based. Many of our controls are usually implemented by public or other institutions for the duration of projects. And of course, when those projects end, the management stops. It's also, well, at the same time, dependent on project funds. And so when funds dry up, usually the institutions that are tasked with the management don't have the resources to be able to continue with the management of the pest. What we are seeing also is control is usually difficult. It's labor intensive or it's expensive. And so it's usually not able to be implemented by farmers, and as a result, our farmers are usually sidelined in managing pests. The second problem we have is replanting. We have, out of those 80 or so million palms, most of them are actually senescing. They are aging populations that need replanting to improve the livelihoods of our communities. But to do this, of course, we are mindful of that pest problems may be aggravated, especially coconut rhinoceros beetle and weevils, for example, and perhaps other beetles. So we need to apply best practice before we proceed with any kind of plan for the replanting. But what is best practice for multiple pests? I mean, here we are trying to address multiple pests. And so we have a dilemma there on what is best practice if we continue down with this kind of management options that we have been seeing for other pests in the region. So coming to our draft action plan, it's very busy, so I'm not going to go through all of it. But basically, this is the draft that we've come up with. We've gone through the theory of change. Just to summarize, we have 4 main outcomes for the theory through the theory of change. These are medium-term outcomes. One is the effective coordination and collaboration in research for priority pests in our Pacific Island countries and territories. The second outcome is to improve the management and management methodologies developed, new management methodologies developed and implemented for priority pests. Outcome 3 is about effective detection and timely containment of pre- and post-border incursions. Alan touched on that in terms of biosecurity responses. And outcome 4 is all about empowering farmers to manage threats through capacity building and holistic farming systems. So the medium-term outcome is an improved preparedness and community adoption of effective integrated pest management farming systems. So watch out for that acronym, IPMFS. So the final impact that we want to see is that there's a reduction, of course, in the impacts of coconut pests in our farming communities, and livelihoods are improved and sustained. So the focus of our inputs are on preparedness, understanding the threats, responding to the threats, new ways of managing the threats, and a participatory approach to pest management. And I just put a note there that all of our priorities are set by Pasifika NiuNet, and all of the projects will be channeled through them. I just want to digress a little bit to tell you that we are aware of the CRB action plan, and this is what what I've seen from the objectives, and some of these are overlapping with what we are doing. So we certainly hope that we can collaborate in some of the activities already taking place. And so we are all about building resilience in the Pacific against major pests. We want to maximize preparedness and response, both pre-border and post-border. We want to prioritize involvement of farmers in the management of pests. We want to implement holistic approaches to pest management based on sound ecological principles in integrated farming systems. We want to include traditional knowledge and practices also in the implementation and research. Improve production and dissemination of existing and new biological control agents, and the application of modern techniques for control of insect populations. I'm not going to elaborate on any of these. There's lots of areas of work and research amongst those broad categories. So basically the project concepts that we have under development in terms of preparedness and response, we are looking at ensuring capacity in all areas and at all levels in terms of recognition of threats and pests and diseases included. a timely response at all levels, including from farmer right up to biosecurity organizations, authorities. Monitoring also at all levels, including farmers, regular dissemination of information. Again, I think this is already happening for CRB, but we want to see how we can perhaps find new ways of hosting and delivering training events and delivering knowledge, and also farmer-to-farmer information exchange. In terms of the management, the project concepts, we want to include a holistic approach to coconut farming, including participatory trials involving all levels of pest management, from detection to control. We want to develop farmer capacity, allow and prepare farmers to adopt an integrated system of farming that minimizes the impact of pests. So then the interventions that they need to use can be minimized as well to control anything that is affecting their system, farming system. And of course, sustainable management of these farming systems to ensure so that there's cost benefit for farmers to sustainably manage pests on their own, as well as income generation at all stages of their replanting. And of course, innovation will come into there. There are, I'm sure, many new ideas that are coming up and will come up in the future. We want to see new ways of exchanging information and alerts to improve preparedness and response. New technologies and ideas to manage pests, of course, including CRB. Introduction of novel new biological control agents. I think that's already happening for CRB from all of the presentations I've heard in the last 2 days. So that's great to hear that. And also new technologies applied to the management of pests. Improved dissemination and application of existing biological controls, including on-farm and within-country propagation of biological control agents. So we really do want to have a lot more in-country production of these agents and if possible, on-farm production of these as well. So in terms of preparedness and response, we are planning for the delimiting surveys for the 5 major pests. Of course, we can't do every country in the region at this present time, so we are targeting countries in the Northwest Pacific, who's possibly where the pre-border concerns are as well. And we will be speaking to a few of our technical working group members from those countries to see who might be involved in that work, in that initial work. The identification of pests, of course, as well, will be taking place during those surveys. Also, during that time, we want to combine an assessment of preparedness. I believe there's already something going on in that area from a talk yesterday about biosecurity, authorities, collaborations, etc. So hopefully we can link into that to assess and don't need to repeat that kind of work. Identification, of course, of biological controls present in country. We hope that can be also done, combined with the surveys, if possible, to look for beneficial insects as well, and of course assessing farmer awareness and capacity in our countries as well. So for project concept 2, improvements in biological control of the major pests, we want to, of course, identify novel methods of storage and application of biological control agents, whether they be insects or fungi or nematodes or whatever. We want to identify new methods for the control of insect populations, preferably non-chemical. We do not advocate chemical use. We consume a lot of coconuts, so that's just out of the question. Provide support for the introduction and use of biological control agents, provide support to our local entities and our farmers. Build farmer capacity, of course, for the production where appropriate. And identify efficient and sustainable ways to produce these biological controls. And this information will also come from the delimiting surveys, we hope, if we can identify some of the beneficial insects and fungi during those surveys. Under project concept 3, this is quite a large area that will be researched, we hope. We want to look at holistic approaches to pest control through sustainable farming practices. This is actually a picture of in Madang of intercropping with mangosteen at KIK in Madang, Alan's organization. So we want the pest management to be integrated with as much as possible ecologically sound farming systems. Of course, it can't be perfect because it's an agricultural system, but as much as possible, we want to combine a balanced farming system. We want farm level management if possible, and we would be pushing for farm level management for major pests. And if we can do this through an integrated farming system as well, that would also be an ideal situation. And of course, intercropping at replanting would need to be with edibles and not edibles, depending on the beneficial insects and the plants that they require as well. So we're looking at a holistic approach to the replanting, and hoping that we can put some trials in selected countries initially to see how the pests can be managed in those countries. So we have identified lots of research topics, and a lot of this is perhaps already going on in CRB, and we are looking for collaborations with anyone who would like to join us in these projects to research a lot of the activities. We're also encouraging our researchers within the region and taking on students to complete some of these research activities identified under the project concepts. They're actually concepts at the moment. We haven't developed them into full projects as yet. And so there's under concept 1, we've identified areas of pest biology, of course, pest identification, including using molecular techniques and barcoding, repositories for data, identification of biological control agents, and of course, improving farmer communication and reporting, and using new technologies for farmers to communicate with different levels of governance and between themselves. In concept 2, research areas, of course, are production and storage of biological control agents, trials on new methods of dissemination in our mostly smallholder farms, identification of beneficial plants for our biological control agents if they are identified. Of course, processing of waste to improve field sanitation, beg your pardon, and farmer capacity building as well under concept 2. And concept 3, of course, trials, planting designs, identification of crops and intercropping systems, utilization of wastes, on-farm biocontrol as well, production, and value adding and income assessments for our farmers as well. And again, building capacity for not only extension personnel, but farmers. So the way forward, we hope that we can finish final consultations on the project developments by Q1 next year. We will start off with concept 1, which is the delimiting surveys and the biosecurity preparedness and response. Hopefully we can start looking for resources by Q2 when the project proposals have been fully developed. And we need to consult with our technical working group and our governance board as well on these proposals. Then we are quite ambitious, but perhaps we are hoping to have the start to roll out concept 1 by Q4 26, but it might be Q1 27. And of course, concepts 2 and 3 will begin and or follow concept 1. So it's going to be a staggered implementation of these proposals, provided we get the resource in, of course. And we're not talking about a 5-year project. This is going to be a longer term project. So we're looking at least 15 years to roll out all these concepts and have all the trials done. So we are not thinking short term here. We're thinking long-term sustainable implementation of some of these activities. So It's quite ambitious, but I think with the Pasifika NiuNet board there, I think we'll have continuity of these projects. So we're hoping that whoever wants to collaborate with us, wants to join us, we can work together to resource these activities and help our communities who depend on coconut for their livelihoods. So thank you all. I would like to acknowledge all of the participants at the Coconut Pest Conference, of course, the board of Pasifika NiuNet and the technical working group, and SPC Secretariat, to Mark and Rita are here, and Ramona as well. So our very dynamic Secretariat, thank you very much for all the work this year and in the past years. So thank you.
Thank you very much for that talk. Some very interesting stuff and ambitious project, and looks like there's a lot of opportunity for collaboration. We've got plenty of time for questions, so please put those in the Q&A. Currently, the question in the Q&A is, what countries will this project be in?
Thank you, Keith. Yes, for the delimiting survey, we're thinking that we will just look. I think we selected 3 countries in the north. Rita, are you on board?
Hi, Carmel. Yes, I'm here.
Yeah. Can you remember the countries we selected for in the Northern Pacific for the start of the delimiting surveys?
Based on our initial discussions and the short criteria that we had drafted for us to consider, we were looking at Palau, FSM, and I think it was one of the Polynesian countries in the mix too. I think it was Tuvalu.
Oh, okay, right. Yeah, so those are initial. We haven't talked with our technical working group members from the countries as yet, but yes, we will be hoping to have discussions with them at the end of this month or early next month. The other projects, we haven't identified countries as yet. We will be talking through with our technical working group on that.
Great. There's another question up. Who is providing funding for these projects, and do you have an open call for projects?
We haven't approached any donors as yet. We're still developing the project proposals. I've presented the concepts and what we want to put into those proposals. So by Q1 next year, we should be ready to go to the donors to see who can resource those projects. It's possible they may, because they're quite large projects, it's possible they may be multiple donors for different components of the project concept.
Thank you. And I'm just summarizing your answers in writing for posterity in the Q&A. We have another question in the Q&A. Would Hawaii be eligible to participate in this project?
Absolutely. We welcome collaboration from anyone at this conference. And yes, we would definitely welcome some kind of collaboration, yeah, with Hawaii. I see that you're doing some great work, and yes, we would be wanting to also work with you in the areas that you also started working in.
All right, I think I can guess the answer, but same question for New Caledonia. Collaboration with New Caledonia?
Yes, absolutely. If you are, I think New Caledonia is do we have a rep? No, we don't. I'm not sure if there were a technical working group representative for New Caledonia, but I think there is one. I'll get back to you on that.
I'll leave that question open in case you want to type anything more in there.
Okay.
Aloha Miller says, this is more of a comment, but I've heard more than once during these presentations that replanting coconut trees is an issue. Local small farmers all over our world are giving up. Do you have any commentary on that statement, or do you disagree? Or
yes, I think that is an issue because it's been, yeah, replanting has been an issue because there's not been any options for farmers to to understand how they can replant, basically, and what they can replant with. So, yeah, I mean, based on what we know about the coconut diversity now, I think we can provide more information to farmers to enable them to make informed decisions on what they want to replant in terms of coconut varieties, and then how they can then replant a little better. But yes, that is an issue. That's a lot of farmers are moving away from coconut. But I think in the Pacific, coconut will always be part of community
I'd like
farming.
to introduce our next speaker, Dr. Marcel Jardeleza, a research ecologist with the United States Geological Survey, Pacific Islands Ecosystem Research Center, and postdoctoral, oh, sorry, that was for the wrong person, postdoctoral researcher of the Hilo Cooperative Studies Unit. And her talk is leveraging existing data to guide biosurveillance for CRB. And please take it away, Dr. Jardeleza, whenever you're ready.
Okay, can you hear me?
Yes, we can hear you.
Okay, cool. Okay, let me just, all right. Good afternoon, hafa day, and aloha, everybody. My name is Marcel Jardeleza, and I'm a postdoctoral research analyst with the Hawaiʻi Cooperative Studies Unit at Hilo. And today I'll be giving a talk on a project Dr. Helen and I are working on entitled Leveraging Existing Data to Guide Biosurveillance for Coconut Rhinoceros Beetle, which I'll just call it CRB henceforth. So biosurveillance for invasive species can lead to early detections and allow us to quantify the extent of spread. And this can be accomplished through focused monitoring, such as traps placed in high-risk areas. For example, CRB was first detected in the Waikoloa area in the Big Island of Hawaii in 2023 through a public report. Traps were then placed in high-risk areas to understand the spread of the introduction. And surprisingly, a major CRB spread was not found in Waikoloa, but because additional traps were placed in high-risk areas like the Kona Airport, it led to more CRB finds. So a key challenge in invasions is that rare species are hard to detect, but we want to find a population as early as possible, so we deploy traps. And in the case of CRB, the purpose of these traps is to show us where the beetles are and where management is needed, which leads to key questions like how many traps do we need to find individuals? The CRB response on Oahu has collected approximately 10 years of data using over 3,000 panel traps with around 1,800 traps still currently monitored. In the map here, you can see the extensive network of traps around Oahu that have been surveyed repeatedly and have data on when CRB was detected and not detected. So each dot is a trap, and red dots are traps that have captured CRB in the 3-month period that this map depicts, and then the black dots are traps that have not. So we have all this existing data, but we want to know the efficacy of these traps, like how effective do these traps actually capture CRB? Since the establishment of these traps, we have yet to quantify statistically the probability of trapping CRB when CRB are present, especially when they are present at low abundance. And that's when it's the most important, right? Because it's the beginning of the invasion front, and it's a time we need to mitigate the spread. A tricky part about CRB is that survey methods target different phases of their life cycle. For example, tree surveys on foliage tells us about the adult feeding damage that occurred in the past. Green waste surveys looks for larvae. But most of the trapping typically uses a panel trap, which captures individuals looking to feed, and barrel traps, which capture adults looking to lay eggs. And understanding how well these surveys detect CRB can help us figure out the most effective method for CRB detection. Today, I'll be discussing a multi-year CERDIP-funded project focused on estimating detection probabilities of CRB to inform biosurveillance. And this project I'll talk about the overview. It's broken up into 3 tasks. So in task 1, we'll be modeling spatial and temporal patterns of CRB occupancy and detection in relation to environmental conditions. In task 2, we'll be estimating the probability of detecting CRB via eDNA. And task 1 and 2 will be used to accomplish task 3, which is to determine the sampling intensity for high confidence that CRB would be detected. For task 1, we'll be leveraging existing data from the CRB response team, which includes spatial and temporal patterns to figure out how these are shaped by environmental conditions and to test the efficacy of survey methods. And to complete this task, we will be creating occupancy models. So occupancy models are statistical tools used to estimate the true probability that a species occupies the site even when surveys have imperfect detection. So in these models, we first break our study area into grids. It kind of looks like this. And each grid has a true status of whether the species we are studying is present or absent. And while this can change over long periods of time, like if the species goes extinct, we assume that the changes in survey detection from week to week tells us about the imperfect detection rather than the true change in occupancy. So this just means that, for example, if CRB can be found in a trap in 1 week, but the trap might not capture CRB the following week. But just because the trap doesn't catch CRB every single time we check the traps, it doesn't mean that CRB is gone from the site. So taking account this imperfect detection is important because it prevents us from mistakenly concluding that a species is absent just because it wasn't seen during a survey. So in my example here, I will give kind of like a conceptual glimpse of how these models work. So let's say panel traps were deployed because CRB was reported. So if a trap had CRB, it is marked in black, and if it did not, it's marked in gray. The panel traps were then monitored for 3 weeks, and in week 1, some traps were positive for CRB, which tells us CRB was or is present in the site. We go back to week 2, we see a different pattern of detection. In week 2, there's only 1 trap that captured CRB. And again, in week 3, we see a different pattern of detection. So beetle detection varied each time the traps were checked, but it doesn't mean that the beetle wasn't there when they were not detected. It just means that it wasn't detected during certain surveys. But we know that the species is there because we're still capturing CRB, right? And this is why they use the term imperfect detection. So occupancy models uses the patterns of detection and non-detection across repeated surveys to estimate the detection probability. So the fewer traps with no captures mixed in with the traps that have captures increases its detection probability. So occupancy models are just extensions of logistic regressions, if you're familiar with that, except it's specifically designed to address the problem of imperfect detection. And occupancy models are pretty common. They're used a lot in conservation, especially in declining populations of endangered species that are notoriously difficult to detect. So in the case of declining species, if programs did not take account the imperfect detections that occur, we can actually underestimate the population size and the extent of species declines. In the case of invasive species, occupancy models are critical for early detection and long-term monitoring, especially when they are still at low densities and newly invaded spaces like the Big Island example I provided earlier. Occupancy models can also be used to compare trap efficacy. So in our conceptual example here, let's say we deployed barrel traps as well. As you can see, some barrel traps did not capture CRB, but some did. And let's say we checked the traps again for 3 weeks and saw that the panel traps overall collected more CRB compared to barrel traps. So from the number of CRB collected, we might assume that panel traps are better at collecting CRB, right? But that may not necessarily be the case. Comparing traps based only on total captures can be misleading because the environmental context where the traps are placed can have a significant effect on what the traps capture. So for example, maybe the panel traps were placed in areas where CRB are known to thrive, right? So maybe this area had a golf course, which may explain the higher rates of captures in this area. But because we placed more panel traps here, based on the numbers alone, it seems like panel traps are more effective than barrel traps. So occupancy models allows us to include the environmental context in which the traps are placed, which helps us more accurately explain the detection patterns that we see. So there's a lot of cool things that we can do with occupancy models, and the extensive network of traps and repeated surveys done by the CRB response team is exactly the kind of data that we can run occupancy models on. And this is kind of how the modeling looks in real life. So in our first analysis, we will be comparing panel versus barrel traps. So for example, here we have a map of Pearl City area in Oahu, and it's broken up into grids. And in the grids, you could see different dots, and you could see there's a light blue dot, and that represents the barrel traps. And then the pink dots represents the panel traps. And for each cell that has both panel and barrel traps like this one, we'll model the patterns of detection across repeated surveys to estimate the effectiveness of the traps. And as we build on our models, we'll eventually add environmental context like proximity to golf courses or airports. By understanding the variation and detection through occupancy models, we can ask the following questions like, how do environmental conditions affect trap success? Does weather matter? Does seasonality matter? For example, are we more likely to detect CRB during the dry summer months? How does trap placement affect detection? Do distance to breeding sites matter? Do land cover matter? Are traps more effective near patches of coconut trees? And then lastly, what methods are the most effective? Is it panel traps, barrel traps, mulch surveys, palm surveys, or eDNA? Which brings us to our next task, where we will be estimating the probability of detecting CRB via environmental DNA or eDNA. So the development of eDNA for sampling for biosurveillance is really cool because it can allow for surveillance at sites without ongoing lure-based trapping efforts, which is what a lot of the traps are. And this is a great alternative because some sites may be difficult to regularly access, or there might be specific ways these tools can complement each other. So for example, not all beetles attracted to the panel traps actually get captured. They may land on a nearby tree, which we can determine using genetic tools. eDNA is shed by animals into the environment, and our collaborators, the Lockwood Lab from Rutgers University, has found that CRB can be detected via surface eDNA off the trunks of palm trees. And they did this by capturing eDNA shed on palm tree trunks using a paint roller, like in the picture before. And the collected eDNA is then suspended in water, filtered, and then the sample gets processed in the lab using qPCR, which is just a lab technique that measures a targeted DNA molecule. And amplifies it, and then allows us to see if CRB was detected or not. So by extending the Lockwood Lab's previous work, we can figure out how eDNA can complement physical traps. Through occupancy models, we can ask, what is the probability of detecting eDNA on a palm trunk given that CRB are present? We can also improve detection capabilities by seeing how we can efficiently combine trapping and eDNA surveys. And then lastly, we can test the efficacy of eDNA samples at sites with low abundance. And finally, based on our detection estimates from panel traps and eDNA, we will determine the sampling intensity needed for high confidence that CRB would be detected. So here we have Hilo Airport in the Big Island of Hawaii, and the points shown are panel traps. So far, there have been no detections around Hilo, but a key question is, and it's probably an important question for everybody, is how many traps do we need to be confident that CRB would have been captured even at low abundance? So quantifying the sampling effort needed to have a high probability of detecting a small population of CRB enables the development of a model-informed biosurveillance strategies that can be applied throughout the Indo-Pacific and could really benefit the many communities that CRB affects. And we just started our first year of our project, and we'll be focusing on task 1. And we're excited to get started on this work, and we'll hopefully have cool results by the next CRB Symposium, which hopefully is in person, which will be really cool. Thank you, Sejusma'asi Mahalo, to our funding and partners, and I will now answer questions if time permits. And my email is below if you're interested in our work.
Thank you so much, Marcel. I'm looking for questions in the chat and in the Q&A. I see one in the Q&A. First of all, yay, detection probability. I taught population ecology for many years, and it was one of the things I had to mention over and over and over again. Okay, what creatures are you finding in your bycatch?
In our bycatch, I'm not the best person to answer that because mine's literally just ones and zeros, whether we catch CRB or not. So I think that's probably a better question for the CRB response team than myself. But yeah, that would be interesting to know.
Do you know? Sorry?
I don't know if anybody else knows that.
I think any of our presenters or hosts can also answer questions in the Q&A. So yeah, maybe we'll see. Looks like one of them is putting in an answer right now. Do you have any sense of how long eDNA can persist and be detectable?
Oh, that's a great question. I'm not sure as well. The Lockwood Labs is the one that's like the main people for that. And actually they did a really great talk last year. So you could watch in the old symposium from last year, which might answer that. I'm really just the numbers person, the person that's doing all the computer work.
All right, there are a number of others you can answer in the Q&A panel, but there's one more that I'm super curious about that I know a ton of people are also very, very curious about. And I don't know how hard it would be to answer it, or if you are even working on something that could lead to an answer to this one. But can CRB traps attract CRB to an area that might not otherwise come to that area?
That's a great question. And I think it was answered by Keith yesterday as well. And I think I am not the best person to answer it thoroughly, but basically he said not necessarily and no. But yeah, I don't think that the traps attract them enough like other things like the coconut trees and stuff, like rotting stuff.
Okay, great. Well, thank you so much for sharing your research. And there are more questions in both the chat and the Q&A panel. Yeah, we'll be excited to see a follow-up presentation from you next year.
Thank you.
Thank you so much. Dr. Daniel Jenkins is a professor at the University of Hawaiʻi. The title of his talk is Precision Aerial Pesticide Application for CRB Control. And yeah, Dan, take it away whenever you're ready.
Okay, thank you. Let's see, I'm trying to share. Okay, share screen. Whoop, share. Sorry, muttering to myself because I can't multitask. Okay, and then hide this. Okay, I think I'm gonna get through this really quickly. My apologies to everybody and especially the organizers. I made your life difficult. I wrote down the wrong day on my calendar, so I spaced out and was checked out doing something else when I allotted time slots. So I really appreciate giving me a little bit of time to kind of share with you. I'll go pretty quickly. It's pretty straightforward. I'm just presenting about kind of drone applications of pesticides into palm crowns, primarily coconut palms. So I'll kind of explain some of the background and rationale. I don't think I need to elaborate too much on that. I mean, most people here are very familiar with this problem, and kind of describe again the hardware we're using and kind of our experimental approach and some of the results. And then some of the highlights of the outcomes in terms of addressing new incipient invasions on other islands. Again, I think most of you are aware that it's kind of gradually, in the last couple of years moved from Oahu and the Hawaiian Islands to other islands. So again, this slide is nothing that you guys haven't seen before. So I'm probably just going to move on here. So our primary interests, again, one of the early control efforts, and again, thanks to Jichung Chung, who presented on Tuesday about some of the chemical methods of control, is basically trunk injections of imidacloprid and other chemicals. And it's kind of become a standardized approach for kind of managing some of the adult beetles populations on Oahu. And so, again, the idea was to try to kind of use a contact insecticide directly applied in the palm crown, again, to accelerate the actual effectiveness, and then also kind of mitigate the kind of spread of the insecticide throughout the palm tissues, as kind of again, as a supplemental or an alternative treatment with this trunk injections. So, whoop, not in, okay, here we go. There's the, sorry. I don't know if hopefully people are seeing my screen. So the platform we're using, again, just a drone. So the experiments that we did, most of our kind of initial experimental trials, this was a few years ago now, was an American-made drone. It's a small one. I mean, the industry is tending to go much larger and kind of rotary atomizers, which is really not appropriate for this particular application. But again, we're using an aircraft called Precision Vision 22X from Leading Edge Aerial Technologies. This was our first aircraft. And essentially, again, sort of following roughly kind of the labels for a product called Demon Max, which is labeled for use in kind of boring insects and termites and things and wood products. The idea is basically to drench the crown itself to get a good thorough wetting and hopefully pull up, the material in the notches and so on, so you get a good dose, but not have it run off. And so kind of using those criteria, roughly it's about a half-gallon crown drench, again, is applied directly and precisely into the crown. I mean, any spray on the palm fronds is obviously wasted and not doing anything useful. So again, try to maintain that precision application right in the palm crown. This is actually a video of a different drone. This is a Chinese-made drone, really nice and compact, has a lot of nice features that, for obstacle avoidance and so on, making it safer and so on. You can, if you were paying attention to the video on the kind of upper right, again, we're using a orifice plate nozzle, and basically applying a kind of a steady stream right into the crown and sort of painting it around in that area to get a good coating right into the palm crown and the kind of notches of the fronds. So we tried several different products, primarily Demon Max. So again, the active ingredient is cypermethrin, diluted it to 0.5% active ingredient, and again, applied half a gallon to each tree. All of these products we applied at, at 0.5 gallon in terms of the volume in each crown. So we also tried a so-called, organically labeled, a product that's labeled for organic operations. So basically a pyrethrum, just a concentrate extract from plant materials, called Evergreen. And then we also tried Mustang Max, which is labeled in the US for agricultural use on tree nuts, including coconuts. The disadvantage of this one for many applicators is a restricted use pesticide, which can, all of these have different issues related to kind of labeling and the allowable uses in the United States. So all these experiments, we had the fruit and flowers removed so that we're not kind of applying onto those materials where pollinators would be kind of congregating and having their activity. So we did, again, these experiments that I'm describing here were done in 2023. So the first kind of trials we did at Hawaiʻi Country Club. And that first video I showed in my presentation here kind of shows a year after, some of those trees that have been completely defoliated. But, we kind of did some preliminary experiments before they got completely defoliated. Again, we picked areas where there were active, kind of a lot of CRB activity as determined by the kind of trapping activity, but from the CRB response. We also kind of did some experimental treatment at Ted Makalena Golf Course on the Waipio Peninsula, directly abutting Pearl Harbor. So some experimental results, I'll go through this really quickly. And so, okay, so if we just look at mortalities, I mean, one of the nice thing about the contact insecticide, generally speaking, if we applied it, especially in well-manicured locations like golf courses where, it's easy to find beetles on the ground, if we kind of scouted the area, an hour or two after the applications, I mean, we would consistently find in the heavily infested areas, dead beetles on the ground. So it's kind of a good delineating tool to identify actively infested trees. And then obviously it's kind of a direct indication that it is kind of a lethal application of the pesticide and killing adults in the tree. So generally speaking, the text here that's in black, I think is kind of comparable, at least at the Ted Makalena. We did all two of the products, the Demon Max and Evergreen, and then control trees with no application. So we never saw any dead beetles that were freshly killed from, not untreated trees, but consistently found dead beetles and Demon Max and Evergreen. And then, around similar times in similar locations, Demon Max was significantly more effective at killing. I have Mustang Max in red here. We went back and treated some trees at Hawaiʻi Country Club with Mustang Max a year later. And I think, it's essentially the same active ingredient and actually more dilute in the kind of Mustang Max treatments. So the reason there's more mortality per tree in that case, again, it's just anecdotal data. I mean, very likely it's just very heavy population pressures at that specific time. We went back, 3 months later, again, just to see how effective these applications were in arresting new damage, so every treated tree. And again, we didn't really take any quantitative data, unfortunately, but, if all of the trees we did before, near the treatment time, and then 3 months later, there's, didn't notice very little new damage, which was good. And then kind of, if you looked at trap catches in the vicinity of, trees that were treated, you kind of see, hard to see, but generally, kind of diminishing trap finds adjacent to the treated trees. So in terms of, practical outcomes of this, we use some of this data to get an emergency use exemption to use Demon Max for coconut palm applications for controlling CRB. It was granted for the outer islands where we have much younger stage invasions ongoing and still not allowed for use under this exemption on Oahu, because it's kind of, I guess, probably as everybody here knows, it's kind of gotten everywhere on Oahu at this stage. There are other products. I think, our Department of Agriculture has approved OneGuard for a lot of landscaping applications and more ornamental palms. And we have active operations and pilots, private commercial applicators using that product in a lot of cases where landowners and homeowners are interested in having that protection. Recently, we've been very active doing, we kind of early stage invasion in Kona on the Big Island at a kind of a new development called Kohana-iki. So we actually had 2 drones operating at the same time, again, trying to treat all the trees in this early stage invasion as part of a coordinated effort to kind of contain the beetle. One success story, and again, I want to emphasize as part of a comprehensive control strategy, there have been outbreaks reported in all of the main outer islands, including Maui, within the last couple of years. So Maui had some finds in Kihei, so it's kind of a little resort town on the kind of south end of the island in late 2023. So we had, we didn't apply it directly in our kind of pilots, but some of our partners from other islands kind of used our approach as part of this coordinated effort to address this new finds in Kihei, Maui in late 2023. And as of now, there still hasn't been any new finds in Maui. So again, it's been effective as part of a coordinated approach. And they were very aggressive in cutting down trees and burning stumps and grinding and so on. I want to give special recognition to Roberto Rodriguez. He's one of my former students that kind of got me into more of the kind of ecological kind of applications for these kinds of things. He's the one that actually did the applications on Maui. He's actually the aviation science faculty member at University of Hawaii Hilo now. And here's some of his background work that he was doing with us. And I think I got through this in 10 minutes, so happy to entertain questions. Again, I really appreciate you squeezing me in, and if there's no time for questions, that's okay too.
Thank you so much, Dan. Great to get the update. We do have we'll do one question, and then there might be another that you can check out and answer in the Q&A panel afterwards.
Sure. Okay.
Could you explain the benefit of rotating between crown spraying and pesticide injection?
Yeah. So I mean, one is the trunk injection, or even I think you can have root drenches in certain scenarios. I mean, it just takes time for the plant to absorb the material and actually, transport it to the actual tissues that are actually getting fed on by the CRB. So there's a delay in the response, and then obviously, as a mechanism to kind of prevent resistance, kind of cycling between different pesticides is generally a good practice.
Okay, great. Thank you so much, Dr. Jenkins. And a few more questions have come into that Q&A panel.
I'll put,
So
okay.
Thank you so much.
Thanks. Yeah.
We're going to go into our next talk. I'd like to introduce Tobias Kohler, who's the director of the South Shore Gardens National Tropical Botanical Garden, and Allerton Garden and McBride Garden. His talk, Beyond Coconut CRB Management Strategies for Conservation Collections, Perspectives from a Botanical Garden. And please take it away whenever you're ready.
All right, thank you, Elliot. Thank you, organizers, for allowing us the opportunity to speak on this. I think that the talk I'm about to give is a little bit different than what we've been hearing for the past few days, in that it's kind of a case study where we take all this energy, lessons learned, science, and we apply it in a very focused, high-intensity effort, and I'm going to talk a lot about a native fan palm called Lolu in Hawaiian. So my name is Tobias Kohler. As Elliot mentioned, I am supported by an amazing team, Michelle Dewey, Leslie Matsumoto, Shauna Walsh, Susan Fawcett and Dustin Wolkes are to name a few. Yeah, we're gonna dive right in here. So we are a network of 5 gardens, 4 of which are in the state of Hawaii, 1 of which is in Florida. We're focused on biocultural conservation, education, research, as stewards of places. I'm building up to the why, right? So we are about plants, people, and places, and that kind of brings us all together in these past few days as well. We maintain conservation collections of native Hawaiian plants and culturally significant plants, also known as heritage plants or canoe plants, several of which are coconut rhinoceros beetle targets, including non-palm species such as ulu, hala, kalo, maya, and ko. And so we have a lot to lose. I'm not trying to diminish coconut by any stretch of the imagination, but I think we can all appreciate that it runs much deeper than that. And today I'm going to spend a lot more time on the native fan palms. Another reason it really matters to us is quietly on this little island of Kauai here on the South Shore, we have a palm collection, a living collection of palms that ranks number 8 in the world against palm collections. And so CRB and any threats to that collection are, of course, they matter to us. Getting just a little bit more specific, we're essentially a lifeboat or a Noah's Ark of sorts for these plants, especially palms. We have about 704 accessioned plant outs of Lolu or Pritchardia palms, representing 25 of the 27 species that are described at this point. I've listed a few other significant species in our collections as well, and take away from this talk today that a deep dive on particular species of concern that and genetic and cultural and other values that could be lost by impacts of coconut rhinoceros beetle could be applied to things like even coconut. But our focus as an institution right now is Lolu. And why all does that matter? Well, on one end, it's about extinction and these plants being so close to the edge. These palms, these Lolu palms, house history and stories that are preserved through mele and chants. And so it just makes it harder to perpetuate those stories and that history in the absence or the threat of the absence of those plants. It goes on to kind of ecological importance, where whole ecosystems and lowlands of many of the main Hawaiian islands and the Northwest Hawaiian Islands had forests of Lolu, and there are mere remnants, tiny remnants remaining today. And then just biologically, in the classic Hawaii, remoteness sense, 23 of the 27 Pritchardia taxa are endemic to Hawaii. And hold on, just gotta get my notes sorted here. 23 species endemic to Hawaii. 18 of those 23 are single island endemics found only on one island. And 6 of those species only have less than 50 individuals in the wild. And NTBG's gardens hold the most comprehensive collection of those Lolu, but some only in very, very numbers. So I reiterate, we're dealing potentially on the cusp of extinction. Lolu is vulnerable to CRB just as palms are, and hoping and learning from the IPM, the integrated pest management strategies and discussions we had for the past few days. We're trying to be out ahead of this and drawing on everybody's experience, research, and insights. So we have real firsthand observations of Waimea Valley on Oahu, which had extensive Lolu collections that are struggling to exist at this time. And then also reports from state partners that coconut rhinoceros beetle has made it into wild and native populations in intact forests. The growing spears of these palms are a little bit smaller, and so they just can't withstand the hammering that the beetle can do. And they're fundamentally way more vulnerable to secondary infections. So you can't get even a bite by a beetle and have them succeed because fungus moves in. In the storage sense, the preservation sense, NTBG's calling is very, very strong. I really just need to emphasize that living collections in this situation are second only to wild populations in importance because other storage techniques or other lifeboats, so to speak, are not easily available or in full action yet. So you can't store seeds. We're working on micropropagation quite successfully, but once it starts to grow, you have to move it someplace, and that all takes a lot of effort. So I hope that answers the why. This is a slide or a map that I think we've seen in different versions over the past few days, and actually at the last symposium. But I just wanted to start where it matters for the island of Kauai. Basically, our start date was March of 2024, so just over a year ago, where our detection started to happen in earnest in the gardens. We managed to only have a whole year go by before a first breeding site detection happened in the valley. And for geographical reference, the yellow dot is NTBG's McBride Garden, Allerton Garden on the island of Kauai, and the entry points and the spread began on the east side of the island. I think one thing we had going for us in the fight against coconut rhinoceros beetle is our inventory system. As a botanical garden, we have an extensive plant records and database where we are almost instantly able to map out our 704 individuals, or just over 700 individuals, and immediately begin to use that information to aid in a prioritization tool. I think, and that basically feeds right into kind of our triaging. We went in working with our on-staff scientists and with our conservation partners to identify, based on rarity, again, agency and research priorities, special projects having to do with pollination and fruit harvesting, and frankly, crown accessibility, which resulted in 4 groupings of palms guiding our plan of attack and management. So group 1 for us is Lolu. There are 704 individuals in the ground on our properties, and we'll talk about what we're doing for those. Group 2 are palms of conservation value, and so we are managing about 50 additional individuals on top of those 704 for that. We have aesthetically and otherwise valuable coconuts, so in our collections, adds 60 there. And then if you look at other target species, we quickly add upwards of hundreds, if not a couple thousand additional individuals to our priority list. And so starting at the top with collections and curation and conservation, it's not just about keeping them alive, although that is a very high priority. We're working on 5 taxa, specifically where we bag inflorescences once they open, and then hand pollinate, and then develop caging fruit. Those immature fruit are often then harvested for embryo extraction. Basically, this is very research driven, and we also then work on where's the material going to come from and where is it going to go. So we have our living collections or ex situ collections, and some wild collecting supported by different grants, and of course agencies helping with permitting and regulatory support and prioritization. Kind of getting into, again, tip of the hat to everybody who's been sharing their Mānoa or their insights and wisdom on how to do this, because it's been quite effective for us. We have 12 traps that are checked weekly. We are detecting about 20 beetles a week on average. A bad week for us is 33. a good week is under 10. We check all of our palms, about 1,200 palms weekly for new palm damage. And then we are checking our mulch pile netting twice a week. And then we have crown netting in place that we are also checking weekly. That is all being fed into spreadsheets, and we check, for example, even whether the beetles are trying to exit the mulch piles or if they're trying to enter the mulch piles to inform breeding site. And I'll get to that a little bit more in a second. When it comes to managing host plants, this is our plan of attack. And again, focusing mainly on Lolu. We are planning and or executing on drone crown treatments every 4 months, or sorry, every 3 months, alternating contact pesticides. We've completed 2 full treatments with our next one coming up next month. This takes us about 5 days to treat about 700 palms under ideal conditions. So kind of following on Dr. Jenkins's presentation right before mine, you'll see this in this photo that we're even treating palms that are at chest height. And that is simply because we can deliver product onto, and for human safety and exposure, we can deliver products so expeditiously. And while it does come at a premium, it's incredibly fast for us. And then we're also, as I mentioned, we're doing some netting the growing spears. We've got about 250 growing spears netted. And then we also do drenching once every 12 months. It takes about 6 people, 5 days to accomplish our drenching undertaking. Either way, this is an enormous amount of effort. And we have to adjust those nets, by the way, every 1 to 3 months. And I mentioned that we check those weekly. When it comes to breeding site management, again, we've taken you guys' lessons learned and deployed it quite fully. We shred our green waste at least once a year at a $30,000 or more expense. We then net our mulch piles. In addition to the netting, we turn them every 2 to 3 months. In terms of using our mulch, what we call fairy dust, we just thinly apply mulch everywhere we go. And the result right now is constructive. We have found little to no emergence from our mulch piles. And since we initiated this effort, we've actually only found 3 grub kind of hot spots, so to speak. And so, again, a big thank you to everybody for giving us the insights to start this early and hit it hard. I think we're, knock on wood, holding off the worst, at least at the moment. And this gives us the breathing room to do the research and development and outreach so that we can bring our neighbors, our communities, and our greater globe, I guess, folks in the loop and to care about it. So we need to know what we're trying to save, and so that we're doing phylogenetic studies. And Dr. Fawcett is leading that effort to make sure that we're truly focusing where we need to. We have a great set of folks, master's students, as well as staff and researchers working on the reproductive biology so that we can speed up and keep these palms productive so that we can get offspring from them. And then we are working, as Dr. Cheng spoke on day 1, on looking at different mixes of injectable chemistries along with Arborjet to try and, again, higher impact, lower dose type, more effective solutions for injections. Let's see, and I think that's getting close to, yeah, and we're always willing to embrace new technologies and be a test site. We've got a lot to lose on this one. And so with that, I just want to thank again, and these folks are actually sitting in a room right next to me right now, for thousands of hours of effort at this point to keep these incredibly rare palms alive. And a huge thank you to everybody literally across the Pacific for this immense amount of knowledge sharing and collaborativeness that makes this possible. And that goes to my next slide, because Hawaii's got one heck of a home team when it comes to making us successful in this effort. And I will yield whatever minutes of time I have back to you, Elliot. Thank you.
Thank you so much, Tobias. Move on to the next presentation. Mark Manglona. Mark is with the United States Department of Agriculture, National Fish Wildlife Foundation, Department of Interior, as well as CNMI, Customs and Biosecurity, and he's here to talk about coconut rhinoceros beetle detection, eradication, and prevention measures on Rota and the Commonwealth of the Northern Mariana Islands. So Mark, I'm going to hand it over to you when you are ready.
All right. Okay. Good afternoon. Good morning, everybody. I'm just gonna update you guys on how we're doing on Rota. So this is my screen. Start slide. You guys can see my screen?
We can. It looks good.
Okay. So this is my presentation since yesterday. So for update for us here in Rota right now is I really appreciate of my team right now, because we are so aggressive in Rota right now since I started working back in 2017 when they first discovered the coconut rhinoceros beetle here at the ground zero at Tewksbury Park, which is right here where they first discovered the Tewksbury coconut rhinoceros beetle. Since then, we've been very aggressive on the coconut rhinoceros beetle, because I really don't want this pest on the island of Rota or anywhere else in the region of the neighboring islands. So we are so glad that we're so aggressive that for 2 years right now, we haven't been catching, once or 3 times a month we've been catching. So I can tell you since January of this year, which is, you can see the chart that I had up, the CRB since 2017 when we first discovered it. And then to now, since January, we only caught 3. But this is only on the traps, the CRB traps and board panels. On February was only 2. And then on March was only 39, which is it's in an infested area where we cut down a dead snag. That's all we collected from that area. So April was only 2. Since May, 16. That's all in the board panel and ground trap nettings that we've been fighting. So as for, where's my map? Sorry guys. Okay, so this is the only the main sites of the CRBs, which is Tewksbury and Paw Hotel. In 2022, we found the new infested site in Con, which is above the Titetu village. And then on 2019, this is the second site that I discovered in 2019 when I did my survey, around the island survey. So since then, we are so aggressive, especially I'm aggressive after working hours. I go out every night, 4 to 5 hours to check all 356 traps. So I'm very glad that the CRBs here on Rota are low. We've been catching it in the board panel and ground net traps. And we do actually need more acquired additional CRB staffing, which is one of them right now. We lost one. I hope he found a new job. So hopefully he has a great year this year. So we lost one team. So there's 4 of us right now, the ground teams. So that's basically it, the update here on Rota, which is sounds good for us right now that it's under contained right now. So that'll be it for me. I can take any questions.
Thank you so much, Mark.
No problem.
Looking for questions in the Zoom panel. This is not a question, but Aloha Miller says, Rota, awesome. I'm gonna look in the chat and give another minute to see if any other questions come in. I guess here's one quick question, and then we'll see if any others come in. Is there any evidence that adult beetles could fly between different islands in the Mariana Islands, or are they just too far away?
Well, from Guam is only what, 3 hours? But since the origin of our CRB here, the coconut rhinoceros beetle, is the same as the one in Hawaii. I'm very surprised that why is not a CRB G, but it's CRB as similar as Hawaii, the Big Islands.
And do you? I'll ask one more, and then we'll move on to the next presentation. So I'm not seeing. Oh, unless this one. Not sure if that was for the last talk though. Maybe, yeah, tell me if this one is relevant for you, Mark. It says, what are you spraying or injecting?
Well, we've been spraying Demomax since 2019, but since the military stepped in and environmental assessment, so we stopped since June of 2022. One of my former coordinators, Frank O'Don, told us to stop spraying because they need to do an environmental assessment report. So since then, since we stopped that, well, man, the coconut trees are so healthy at Tewksbury Park.
All right. Well, thank you so much, Mark. There are a couple other questions in the chat and then in the Q&A panel, so I'm hoping you'll be able to check that out and add in any answers you have.
Okay.
Thank you so much.
No problem.
And we're going to move on to the next presentation. This is Marley Monahan, who is the CRB Response Canine Handler and Trainer for the Kauai CRB Volunteer Program. And she'll be presenting Integrating Detection Dogs into CRB Management and Eradication. And take it away whenever you're ready.
Awesome. Give me 1 second. All right. Hi, everyone. My name is Marley Monahan, and this is Integrating Detection Dogs into CRB Management and Eradication Programs. I have worked with the CRB response as part of the canine team since 2021, and independent of that, I have assisted Conservation Dogs of Hawaii in the training and implementation of volunteer detection dog teams on the island of Kauai. So that said, this presentation will not be about the finer details of dog training, but rather about the various ways detection dogs can contribute to the overall CRB management efforts. So to get started, we can talk about the basic ways detection dogs are helpful. By now, most of you probably know that CRB breed in decomposing plant material, and that means that, so as I was saying, as far as how dogs are helpful in CRB detection. CRB breed and decomposing plant material, which means that in many places there is an overabundance of material available for them to breed in year-round. Research is still ongoing to determine effective methods of CRB control, but a good place to focus efforts is in the removal of CRB larvae from breeding sites prior to their dispersal. So with all of that potential breeding material that accumulates in tropical environments, it can be very difficult to search it all by hand. So by using a dog's sense of smell, we don't necessarily need to sift through every pile of mulch or every rotting stump to target breeding sites. And we can instead cover more ground and identify those breeding sites with a lot less time, labor, and money involved. So to give you an example of what that might look like, I've included a short clip from one of our training sessions on the next slide. We'll see if it works without the optimized video option on. I do apologize, everything worked in our test check, but we will give it a go. So here, this is me with Ryder, one of our dogs from our first cohort of CRB detection dogs. You can see there's mulch underneath all of that vegetation and there's a target hidden somewhere. As he comes around the corner, you'll see he'll catch something and he takes time to really detail the area before narrowing down to the source. And then he alerts to the odor with a sit. So this is just to give you an idea of what this might look like in the field. And it would be the same concept where we'd have a large area of potential breeding material. And if the dogs smell odor, they would let us know by doing their trained response, which for Ryder was a sit behavior. So a common question we get is about what we look for in the dogs we select and train to do this work. And although breed can affect suitability, it's not the most important thing we look for. Mostly we're looking for dogs that are highly motivated by food or toy rewards, and then we use those rewards to condition the dogs to value CRB odor. So it's also important that they have appropriate characteristics for the particular job we want them to do. So we might not necessarily want a loud, high-intensity Mānoa searching unsuspecting travelers for citrus in the airport, but they might work very well in an open outdoor environment when hunting for CRB. Now, how a program incorporates detection dogs might vary depending on the particular goals and structure of that program. For example, the CRB response is a state-run program within the University of Hawaiʻi system. That program is broadly focused on managing the impact of CRB in Hawaiʻi, and the canine team is only one small part of that overall project. The canine teams specifically are directed by the USDA, with all of our dogs coming from the National Detector Dog Training Center. So both our dogs and our handlers are trained, certified, and held to standards set by the USDA. On the other hand, we also have organizations like Conservation Dogs of Hawaii doing similar work, but with volunteer teams. CDH is a nonprofit focused on training and deploying detection dogs within a variety of conservation projects across the state. So while their main focus is not CRB specifically, their focus is more conservation detection dog work generally. And earlier this year, they did launch their first CRB detection dog program on the island of Kauai with 6 dogs and their handlers. Those teams are still in the process of training, but are expected to be up and running by the end of the year. So in many ways, the work being done by these programs is very similar, but the way they structure and approach the work can be very different. So for example, we hope the volunteer teams will be particularly helpful on Kauai. By the time CRB were discovered there, populations were already pretty well established across the island. And around the same time, early detections were made on the Big Island and Maui. So the decision was made to focus more resources on those 2 islands where eradication was still considered feasible. So the CRB response, while based on Oahu, is a statewide government program that has to spread its resources broadly. Meanwhile, the volunteer teams can offer additional resources to the communities on Kauai where eradication may no longer be the primary goal, but that could still benefit from continued detection and additional control measures locally. So if you do have a CRB program that is interested in utilizing detection dogs, it is important to consider the program's ultimate goals and structure, as well as the current status of CRB in that area before trying to incorporate a detection dog program. Then the ways that dog tames are applied may also change over time depending on how program goals change. So for the CRB response, focus has shifted over the past 5 years from complete eradication when CRB was only present on Oahu, in which we worked with the dogs to identify as many breeding sites as possible. And it has since shifted more towards containment as we have seen it spread to different islands. In the past year or so, we've also broadened our work to assist in early detection efforts on neighbor islands. So, for example, early on, CRB populations were lower on Oahu, so we deployed the dogs in areas along the borders of known infestations. So if we see these being the general hotspots on Oahu, this was back in 2021, 2022, we kind of focused dogs on the outskirts of these moving outwards so that we could try to curb further spread. And then we would also focus canine surveys in areas where new detections or changing detections were made, like, for example, through catches in pheromone traps. However, more recently, CRB has become established pretty much all across Oahu. You can see this more recent map that shows overall trap catches in different parts of the island. So we no longer have the resources to search all potential breeding material on the island. So instead, we have to focus our survey structure much more narrowly, and it is now confined to more of a 250 or 2,500 meter zone around all airports, harbors, as well as properties that are shipping potential breeding material such as compost or potted plants. So if we look at this map here, this outlines those buffer zones around those ports and harbors. And then we also have several areas that we focus in the central part of the island and on the windward side where there is an abundance of nurseries and other green waste processors that we're focused on. So detection dog teams can be applied in many ways and can look very different depending on program needs. But in our experience, there are a few ways that they're most beneficial. And generally, dogs are going to be most helpful in places where there is a lot of potential breeding material to search, especially when CRB populations are still low. The denser and more widespread a population, the easier they generally are to find using other methods. So dogs help, especially so when it's more of a needle in a haystack situation. And this is especially true with early detections and rapid response efforts. And then they're also more effectively applied when you have a clear place to start. So if you have a way to monitor CRB populations and changes like we do with the pheromone traps, that can then inform a good starting point where the dogs are deployed in the field rather than just taking a shot in the dark. That said, there are a few limitations to consider. Dogs are not robots, and they do have an incredible sense of smell, but they do have limitations. CRB is particularly difficult odor, even for experienced teams, especially considering how deeply it is often buried. So if the odor isn't accessible, the dogs can't alert to it, and it is not uncommon to see environmental factors like wind, temperature, and moisture affect how easily the dogs can detect breeding sites. Additionally, because there's often such a large volume of material to sort through, even when the dogs have pinpointed, verification can be very difficult, and it's not unusual for us to have to come back days, weeks, sometimes even months later before we actually find any CRB in the area they alerted to. So we do generally schedule repeat surveys in these sorts of situations to ensure that nothing goes missed just because we weren't able to verify with our human hands and human eyes. So even with these limitations, the detection dogs can be a huge asset for CRB management programs. So if we look at some of the metrics from the CRB response canine team, so far this year in 2025, we have identified 30 new breeding sites within that buffer zone around those high-risk ports and nurseries. So that was that map I showed you a few slides ago. This also includes the early detection of the first breeding site found in Kona, which was only the second site to ever be found on the Big Island overall. So I think this is a good example of how even within these limited search parameters, the dogs are still capable of detecting CRB. And even when we have those narrow search parameters, there may be fewer detections, but the sites we do identify in those cases are especially significant and impactful. So like that early detection in Kona was a huge break in terms of trying to get that under control before it really gets established. The dogs have also been conducting regular inspections of potted plants and green waste that's headed off island, clearing them prior to transport, and have also participated in several educational and outreach events. Additionally, while the CDH teams are not fully up to and running yet, they have already identified over 20 new CRB breeding sites as part of their field training in the more heavily infested parts of Kauai. So once they're up and running and actually out doing systematic surveys, we're hoping to see a whole lot of success from them as well. So just to summarize, scent detection dogs can be highly beneficial to a CRB management program when designed thoughtfully and employed appropriately, but their greatest benefits will be seen as part of rapid response efforts, early detections, and routine biosecurity clearances. So again, they can be a huge asset. Just make sure that you do so with intent and think about what you need in your own program before just bringing dogs in to help. So if you would like to learn more, here are the contact information for the CRB response as well as Conservation Dogs Hawaii. You can find them through these websites or social media channels, as well as email or phone. So if you have any additional questions, feel free to reach out. And with that, I would like to thank you all for joining and bearing with my technical issues. I would be happy to take any questions at this time.
Thank you so much, Marley. We've got a few questions in the Q&A panel and in the chat. And maybe we can do one live, and then if you don't mind taking the other couple afterwards. Can you share your thoughts on how close you think Hawaii is to having the number of detection dogs and handlers that we really need?
That's going to be, I think that's going to be a hard thing to quantify, like how many we truly need. I think we are at a good start because in addition to the CRB response, so we currently have 2 handlers on Oahu, but we also do inter-island work occasionally. And then we also have the 6 volunteer teams on Kauai. And there are also some smaller smaller businesses that have dogs as well. Like I know there's one on Big Island currently. But additional detection will certainly be helpful. And I think that it only becomes effective if there are also resources in place to then follow through with what the dogs find. So before jumping the gun and saying we need more dogs, we want to make sure we also have the infrastructure in place to then follow up with those responses and those identifications. Otherwise, we're just detecting material with not much to do about it. So at this point, I think it's good. It's still a little bit experimental in some places, trying to see how much of an impact it really can have. But part of that is really going to hinge on what the follow through will be.
Great, thank you so much. Really appreciate it. And yeah, we do have a couple more in the chat and Q&A. So thanks so much for sharing about your work.
Yeah, thank you.
And our last presentation in this detection monitoring session is Dr. and Emeritus Professor of Entomology at the University of Guam, Aubrey Moore, who will be presenting Improving an AI Model for Automated Roadside CRB Damage Surveys. And Aubrey presented last year, so it's great to have you back. And whenever you're ready, please take it away.
Can you hear me?
Yes, we can hear you.
Oh, fantastic. Hafaday, aloha, and mayang buntag from the Philippines Islands. Sorry, wrong screen. Can you see the V-shaped cuts?
Yes.
Fantastic. Everybody should know what they look like by now. We're kind of fortunate having these V-shaped cuts because they're very specific for CRB damage, and they're very noticeable. They can be seen from over half a mile. We got to remember that these V-shaped cuts were only made by adults. the larvae don't do any damage at all. It's the adults that bore into coconut trees to tank up on high octane fuel on the sap for flying around. So these V-shaped cuts were made probably a month or so before that frond emerged, when the beetle was boring through the outside developing leaves into the white meat to suck up the sap. So it's kind of incidental damage. They actually don't feed on the leaves. They macerate the leaf material and then suck up the juice that comes out of it. So seeing V-shaped cuts in more advanced damage is a little harder because everything gets all kind of confused up. These plants here are not dead. They're not really dying. They could be saved if you could stop feeding by using pesticides, for instance. If you were to spray these trees every 2 weeks with cypermethrin, you could save them. You could bring them back to apparent full health in about 15 months. It's a big job and it's expensive. However, the dead one in the middle there isn't going to survive. It's lost its capability to produce a new spear. The meristem has been damaged. This only usually happens when there's a high population and that the tree has been attacked simultaneously several times. So why do we need standardized CRB damage surveys? It's for monitoring changes over time and space, especially in response to control activities. You want to know if the population is going up or down and if it's spreading. And also for detection in CRB in new geographic areas. If you want to read a really good article on damage assessment for CRB, read this paper that came out written by Sarah Mansfield and her colleagues at Ag Research New Zealand. It's really excellent. So what they suggest is using a point system for ranking each coconut tree. The simplest one is just damaged or undamaged. Then there's a 3-point scale: damaged, undamaged, and dead. And then a 5-point scale, which is a little more complicated. In addition to this, it's also good to count the number of V-shaped cuts in each palm. So data acquisition, you have several choices. You can just drive around and do direct visual observations, write down your results, or put them on some kind of a data logger. You can use videos and still images and then sit in a nice air-conditioned lab and review your data. Or you can do ground-based and aerial imagery for large surveys and again, analyze the data in a nice air-conditioned lab. My idea was to use a cell phone to drive all the major routes on an island to do a very rapid island-wide survey looking for CRB damage. So this is my first attempt at that. There's a cell phone there. It has a free app in it called Open Camera, which is programmed to take a picture once a second. And it also logs the GPS coordinates using the GPS chip in the phone and stores those embedded in each picture. You don't need cellular access to do this. Everything happens in the phone. We mount the camera on the outside of the vehicle with a magnetic mount, and the camera is pointed to the passenger side of the road at a 45-degree angle to direction of travel. And it's tilted up a little bit by 15 degrees to look at the tops of the trees. So what do you do with all these pictures that you take? Well, I developed a data processing system which uses exclusively free and open source software. So you take the images, you run them through these AI object detectors. The object detector finds every coconut palm in every image and ranks it as to damage, and it also finds the V-shaped cuts. This goes into a little tiny database called Spatial Light, and this automatically is pumped into a GIS system. I'm using Quantum GIS, which is free. And what comes out the other end is a nice map of the survey showing you the results. And these maps are interactive. You can zoom in and pick certain layers and do all kinds of fancy stuff like that. And these maps can be put up on the internet very easily. So here's an example one. This is a still example one. This was our first island-wide survey on Guam using this system. And at that time, we were using videos instead of still images. And we collected 882,000 frames, ridiculous amount of data. I won't do that again. So that's why we switched to using still frames. So we had the position of V-shaped cuts, ranking of every tree, and we could calculate the percent damage of the trees that way, island-wide, and map everything out. So you can zoom in on the interactive map, and these points are actually the points where each shot was made, where each frame was made, and the color of the dots shows you the damage level. In this particular case, we have an area called Pacific Royal Palms on the left-hand side of the screen. On the right-hand side of the screen is a residential area. Royal Palms is an abandoned housing development that the DOD was running, and they had almost 100% mortality on there, and a lot of dead trees. That's why I have all those red dots. On the other side, in the local neighborhood, most of the trees were healthy. So you can see the V-shaped cuts as well. Those are the purple dots. And when you're finished, you can actually stitch things back together. So this is going down the road that was in that close-up picture there. There's a water tower. Things will start to happen in a minute here. Okay, here's some trees coming up, dead ones. There's the Royal Palms entrance. You get the idea. It puts bounding boxes around the trees, gives them a damage rating, and also puts little polygons around the V-shaped cuts. So we did 10 maps, 10 surveys using this method between 2020 and 23. And we could use the data to plot out what was happening in time as well as in space. So here's what's happening in time. The percent of trees damaged island-wide fluctuates quite a bit between about 5% and 22%. So that's about all I'm going to do on the island-wide surveys on Guam. This same system can be used for other purposes, for detection surveys, that kind of thing, delimiting surveys. And we did this on Rota, which is the island only 35 miles north of Guam. They got infested with rhino beetle. And Mark Manglona was just on giving a really good talk about that. And he was talking about it yesterday as well. So we sent the camera up to Mark, just the cell phone. We sent the cell phone and the magnetic mount, and showed him how to set it up and everything. And he did a survey up there and sent all the data back inside the camera, inside the cell phone. And we actually found 2 signs of coconut CRB damage up in Rota. Here's one of them. I reported these 2 observations back to Mark using iNaturalist. And I just noticed when I was putting this talk together, we missed this dead one here in the middle of the screen. There's a dead coconut. How could the AI miss that? Well, it needs to be trained a little better. We also did the same type of thing on Majuro, which is in the Marshall Islands. Majuro was hit with rhino beetle. First one was detected there October 2023. So I sent over my technician, Chris Kyanan, with a cell phone. He got in a car over there, started up on the east side of the island, which is Rita, little village up here. Drove all the way down to Laura, turned around, and all the way back. Yeah. Took him 4 hours to do a complete survey. And at first we were getting all kinds of hits, but by tweaking the threshold for confidence level, we were able to get it down to just 3 hits, 3 V-shaped cuts. and they were all in the same place. Two of the hits were caught driving down, and one of the hits was caught driving back. So that's kind of confirmation that it's doing its job. So if this was a live map, I'd be able to zoom in and show you all good stuff. And you can actually click on one of the camera positions and see the pictures that were taken at that position. I'm a coward. I'm not going to do that because it might mess up my presentation here. So maybe I'll come back to that if I have time. And then there's virtual surveys. You can point the object detectors at images available on the internet. I was really inspired by work that was being done in Christchurch, New Zealand by the ag research folks. They were actually looking at social media posts from all over the world, looking for CRB damage. And for some reason, they had a look at some shots in Mexico and found what looked like CRB damage. So they wrote a nice little paper on this. Just last year, Trevor Jackson went over there and actually collected some CRB, which were identified conclusively in this one area of Mexico. So I thought, well, I'll train my AI to look at iNaturalist pictures of coconut palms. So I did that. It's all automated, and came up with a couple of hits. Here's one of them. It's kind of hard to see the V-shaped cut here. It's got yellow around it. But anyway, the neat thing was when I got the coordinates for this picture, it was right in the same area that Trevor was finding pictures on other social networking applications. So most recently, in the last year, I've been working on redoing the AIs. I think the object detectors can be much improved quite a bit. So I saved all the data from the Guam survey. So I have lots and lots of pictures, thousands, tens of thousands of still images, all HD. So I have lots of data to work with. So the major change is I changed the base model from faster RCNN, which is old school already, and I'm using YOLO, which is you look only once. It's much faster. In fact, it's about 100 times faster to find objects and to train. And then I changed from using bounding boxes, rectangles, to segmentation, which is polygons around objects. So one thing I did almost a year ago now is I put all this stuff on my cell phone, and I was really surprised how fast it was. This is in real time here. Can actually do real-time detection of coconut trees, do the damage assessment, and this particular app is just a demo. It doesn't retain any of the data, but it's just to show that it works. You may have noticed at the end, it was triggering on a couple of banana trees down here, but that can be fixed. So what happens when you apply the object detectors to the old data? Well, here's a picture from one of the roadside surveys on Guam. So here's, sorry. So you can see there's obviously a dead one on the right there, and there's a sick plant in the middle. We call that damaged. And I can actually see one V-shaped cut. And when I apply the object detectors, it finds the V-shaped cut, it finds the damaged plant, and it finds the dead one. It didn't find the V-shaped cut that I was looking at. It found another one. However, my object detectors right now have only been trained on less than 200 images. What you do to train an object detector is you mark up the images, you annotate them by putting boxes or polygons around the objects you're interested in, and then you have the object detector look at a bunch of these examples, and it'll eventually learn. I've only annotated about 200 images. For models of this kind, you usually use at least 1,000, up to tens of thousands of images. So I've got a long way to go, and I think things will improve dramatically over time. So concluding remarks, this really shows that damage surveys using this automated method are much, much cheaper than using Pheromone traps is about 10% of the cost. And there's another reason why you might want to use damage detection instead of pheromone traps, because the relationship between CRB trap catch and CRB population density is actually unknown. What is known about the way pheromones work is that pheromone trap catch actually reduces at very high population densities. And the reason for this is you have a lot of competing natural pheromone sources. In this case, it's the male beetles. So they're putting out plumes, and the plumes from the traps are just getting lost in a big confusion. So the relationship between CRB trap catch and CRB population density, it probably looks like an upturned U. So at low densities, the traps are quite efficient. And then at one point, goes over the top of the U, and the actual trap catch goes down. So if your trap catch goes down, it doesn't necessarily mean that your population is going down. It may mean that your population is going up. I talked about this last year and gave a little tiny 5-minute talk on it, and there's a link here to that talk. So pheromone traps have another problem too. On Guam, we noticed that they weren't very attractive to rhino beetles. We did mark, release, recapture experiments and found out that only 11% of the beetles that we marked, which were released in very large grids of traps, only 11% were recaptured. So for population control, the traps are useless. For monitoring and detection, they're still very useful. But if you have the money, do both, like they're doing in PNG. They have an integrated system where they're looking at damage detection and also trapping with pheromones. Okay, there's also what may be perceived as competition with people that are trying to do aerial surveys to do the same thing. They're looking for V-shaped cuts using drones. There's pluses and minuses to both. Aerial surveys give you precise coordinates for the detections. The roadside trapping only says that there's a picture that was taken here with an object in it, and that object can be some distance away. So it's imprecise ground truthing there. Aerial surveys can't be used in no-fly zones, and there's a lot of no-fly zones in places with military installations. Ground-based surveys can be used anywhere there are roads. Aerial surveys can be used on islands that don't have any roads. Aerial surveys are expensive and high-tech, whereas ground-based surveys, especially using the method that I'm talking about here, is very cheap and low-tech. So all of the data that I collected during the Guam surveys, all the images are available online. The models that I'm currently training are already available online for people to test out. You can actually go to a Colab site and play with your own images. So I'd like to end by acknowledging the Forest Service and Department of the Interior Office of Insular Affairs for funding the initial training of the object detectors, which was done by a company called One Panel. So I'd like to thank Don Sabin and Rush at One Panel. I'd like to especially thank Chris Kainan for doing the 10 surveys on Guam and 1 on Majuro, and for Mark Manglona for doing the survey on Rota. And I'm through. I'm finished. Thank you very much for listening.
Thank you so much, Aubrey. We're running a little bit behind, but I did want to ask one question that's in the chat. Is there an app available yet for this? Like, can people download an app and try to do some of these surveys?
You're talking about the cell phone app?
Yep.
Yeah, well, the cell phone app at this point is just a demo. It's kind of useless because it doesn't retain any data. But I think eventually I'll get back to playing with the cell phone. And what we'll probably do is do a survey and then have the processing done in the cell phone, converting it into a map or something, or maybe just upload the raw data some other place. So in terms of the app, no. But as I mentioned on Colab, I have a Python, a Jupyter notebook where you can actually upload your own images. and detect things in those images. It doesn't work very well yet because the AI object detectors need a lot more training, but you can play with it at least.
Okay, great. Thank you so much for a great presentation. And there might be a question or two in the chat and Q&A to check out and answer afterwards. I bet folks would love to have a link as well to your site so they can check out.
Okay, so that talk I just gave is actually online as an HTML page.
Oh, great. Okay. If you could share that.
I'll put the address to that in the chat.
Okay, thank you so much.
I don't have access to the Q&A for some reason. It disappeared on me.
Okay.
But I'll send it to everybody on the chat.
Thank you.
Thank you very much, Elliot. And thanks for inviting me.
Right, that concludes our session. And we're going to jump right into the next session. I'm going to hand it over to Ritin.
Thank you so much, Elliot. And again, appreciating the earlier presentation from Dr. Moore on the AI. We are now moving into session 4, and this one is insights from the Pacific. If you've been with us since day one, a quick recap of the sessions that we have had already. Session 1, we focused on new technologies, and after that, session 2 on day 1 was on biological control and surveillance, and of course, yesterday was more of regional updates from different parts of the Pacific. We heard from countries in Melanesia, in Micronesia, in Polynesia, as well as a presentation from Southeast Asia, in fact, from Timor-Leste. So I hope that gave you a good gave everyone good insights as to the CRB status in different parts of the region and what each of us are doing in terms of management. So with that, today the session just completed on detection and monitoring, and time now for us to have a look at some more insights from the Pacific. Yeah, and the very first presentation that we have in this session, we only have 3 presentations, so not a lot to go before we wrap up the symposium or the virtual symposium for this year. But this session we are starting with a presentation from Guam, and we are glad that Dr. Glenn Dula is able to join us. Dr. Dula is an assistant professor at the Western Pacific Tropical Research Center, University of Guam, and his presentation will look at the evaluation of long-term efficacy of CRB management strategies that have been applied at Guam's ports of entry. So with that, I hand over to Dr. Dula. The floor is now yours.
Okay, let's see. Thank you for the introduction. Let's what are options? All right, can you guys hear me and everything looks okay?
Yeah, we can see you, Glenn. We can also see the presentation, but perhaps just the way the presentation is set, the format, it's a little bit right in the middle of the screen, dead center. I'm not sure if zooming in is going to help.
I don't know how to fix that. I couldn't figure that out with Elliot on my short tech check.
No worries. It just might be me as well, so we can take it away. Thank you.
Okay. All right. Thank you guys for hosting me today. A lot of great talks so far. So I wanted to talk about, I guess, work we've been doing for the past almost 7 years now. I've been the PI for as management projects around the ports of entry for about 7 years now. First starting with biosecurity at Department of Agriculture, Guam Department of Agriculture, and then now moving on to the University of Guam. So next slide. Okay, there we go. Over the past 7 years, we've been focusing primarily, at least with this project, primarily focusing on the ports of entry. So after CRB was discovered in Guam in 2007, it quickly moved out of quarantine and eradication, and now, it's fairly well established throughout the island, and at least my primary focus when I first joined the Department of Agriculture was to prevent egress out of Guam to other vulnerable islands, especially at the time, even though it's not true anymore. There was a lot of chatter when Hawaii got their coconut rhinoceros beetle, and Guam was sort of implicated in being the source, even though we've been exonerated since then. But still, being the major hub in Micronesia for commerce and air traffic travel, we are a huge threat to the rest of the region. So we have been implementing various management strategies at the ports of entry. So, none of this is new to anybody. We are just implementing the various methods that have been developed from our predecessors. So, brute force sanitation of breeding sites and green waste is by far probably the most effective thing. If we can minimize breeding sites, we can definitely control populations in any area. But being on a tropical island that has an insane amount of annual rainfall and vegetation growth, it's very difficult. Utilize alpha sense, aurictolure, aggregation pheromones, and black panel traps around the ports of entry. We also generate or build artificial breeding sites that we seed with Metarhizium majus spores around the ports of entry. And then also more recently, we've adopted the Hawaii's tree injection protocols using imidacloprid and acephate tree trunk injections, rotating on a 6-month basis. So here's a couple maps of our trap network that we've been maintaining for the past 5 years religiously. We check these traps every 2 weeks, and we also replace the pheromone every 2 weeks just to ensure that we have active pheromone in there. Guam's weather, whether it's dry season or wet season, or whether, depending on the temperature, quickly degrades the aurictolure in the packet sometimes. So despite it saying we can change it every 30 days, we err on the side of safety and just change it every 2 weeks. So the 133 traps we have, we try to encircle the airport, or the Guam International Airport, and then also our only commercial seaport, the Port Authority of Guam. So, even though we've caught, I think it's an impressive number, over 20,000 or about 20,000 CRB adults. As Aubrey just mentioned, they're pretty useless as a form of management, only catching about 10% of the actual population. Catching 20,000 means we missed over 180,000, which is kind of scary. So that's why we implement other management strategies. We use primarily the pheromone trap network to measure the effectiveness of our management strategies. But every beetle matters. So we've been using these pheromone traps to measure primarily initially to determine the risk of the various areas around the ports of entry. So numbered here, 1, 2, 3, and 4 are areas of interest to us. Area 1 is the passenger terminal where all the airplanes load on and off passengers. Number 2 is the integrated air cargo facility where cargo gets placed here and then gets shipped out and packaged to the other islands. Area 3 is our largest airport hangar space run by United, and 4 is our post office, or our main post office. At the port, we have 3 main areas of interest. The actual port facility, Area 1. Area 2 is the harbor of refuge, which local or small businesses run small boating operations in and out of that port. And then number 3 is not a huge threat, is the yacht club, which they do boating activities out of. As you can see in both of the maps, the red circles represent all our individual traps, and the size of the red dot indicates the trap catch rate. So what we notice is, there are, areas of high risk and low risk. So looking at all the traps collectively, we begin to see various trends, which helps us, I guess, focus our management strategies. So looking at it over the 5-year time course, we see that CRB populations peak every year around January. And also throughout the 5-year time course, we have this increasing trend of CRB populations, which, taken firsthand, it doesn't look like any of our management strategies that we initially implemented weren't working very well. But those were, if we break it down by treatment, we start to see other differences. So why do we see this seasonal trend? I'm not a climate scientist by any means. And I tried to, for this presentation, I tried to work in some climate information. I was able to locate one NOAA weather station on Guam that consistently collected rainfall data throughout the 5-year period. And there is only one on the island. Thankfully, it's a NOAA weather station at the international airport. And what it showed me, or what it shows us, is that there's no significant differences between the rainfall. It's maybe except for 2023, it's still not significant. The average monthly rainfall is pretty consistent throughout the years. So that didn't really explain the increasing CRB populations over the years. Overlaying that on our the previous graph, we can see that the CRB populations at the airport, which is the red line, and at the Port Authority, the green line, follow the wet and dry seasons. So after the wet season spikes at around early December, November, then January follows the emergence of all the CRB adults. What we see in this graph also is that we have a trend where the Port Authority, the green line, consistently has lower CRB populations than the airport. And even though I don't have any specific data to support it, I'm just guessing that it's because of, if we look at satellite pictures, we can see that they're in the airport, which is stuck, more centrally, or it's more centrally located in the middle of the island. There's a lot more area for breeding sites and inputs for beetles to come into the area. There's a lot more development, and specifically for the airport, they incorporate growing and planting coconut trees into their landscaping plan. So there's a ton more coconut trees at the airport as opposed to the seaport, which is mostly primarily, surrounded by water, no land, less vegetation, and specifically with the seaport proper, where the containers are loaded on and off, they want zero vegetation to minimize any biosecurity risk with their cargo. So going into our management strategies, we were very, I guess, I don't know how to describe it. We were gifted, or we were granted, a large sum of money by Department of Interior in 2020 to do a large scale, at least large scale for us, breeding site and waste management program at the ports of entry. So at the airport, we were able to remove 126 dead standing palms and 53 stumps. At the seaport, again, they don't actively grow coconut trees, so there were fewer dead standing palms at the seaport. So we only removed 11 there. and we moved about 170 cubic yards of green waste combined from both sites. All the dead standing coconut palms were more or less confirmed to be breeding sites, rotting and full of CRB larvae. So we did this in 2021, and looking at the data for both sites, I'll show this in bar graphs, which is a little bit more easier to understand. In the orange, we have the total, and then in the airport compared to before and after, we have a significant decrease in our trap catches. And in the seaport, which has less coconut trees, less vegetation, it's not really significant. There's a slight decrease, but it wasn't really significant. And you know, we attribute this reduction in trap catch again to the removal of the breeding sites, and then later, fortunately, unfortunately, we were hit in May 2023 with Typhoon Maiwar, which was a Category 4 typhoon, which generated a ton of green waste and damaged a lot of trees, which, as you would think, would generate a lot of breeding sites. Even though the government had a huge program to remove tons and tons of green waste, those were just all from residential sites for the most part. All the unmanaged jungle, you know, all the down vegetation and potential breeding sites were left untouched. So that's what I'm showing here in this graph. We've broken down around the airport our treatments, or the lines are broken down by treatment. Typhoon Maiwar is marked off here with the blue line. We started our GMF metarhizium treatments in December of 2022, and our tree injections in September of 2023. So what I guess what I want you guys to focus on is the trap catches of our control, which is the red line. After Maiwar and before Maiwar, we see this rapid, I guess, spike in populations. And then with our metarhizium and tree injection treatments, you don't see these massive spikes in the populations. A little bit easier to see the before and after are in our bar graphs, which show a significant increase in populations in our negative controls. And then with our GMF and tree injection / GMF treatments, you know, it stays level throughout. You know, I do think I see a more depressed population in the GMF and injection treatments, but my statistics tell me it's not. And that's it. That's all I have for today. So that's quick and we can catch up on time. I'd like to thank all my project leads that have, I guess, put up with me for the past 7 years. My field team, it takes a special type of person to work out in the field and collect beetles and cut down coconut trees. And then also my partners at the University of Guam and the Department of Agriculture. And especially thanks, and you know, this is a huge part of the success of our program, is we've been able to get stable funding from USDA PPQ and USDA Forest Service and Department of Interior over the past 5 years, which really helps keep people paid and motivated to work out in the sun and do, you know, this special kind of work. Thank you.
All right. Thank you so much, Dr. Glendula, for those interesting observations, or should we say updates from Guam. I don't see any questions specifically for you in the Q&A, but there is a comment in the chat box from Dr. Aubrey Moore. So perhaps with that, we thank you, Glenn, for the presentation once again. And we'll move on to the next one. If Alifred Timbola could start screen sharing while I quickly introduce the presentation. Bola is my colleague here at SPC at the Pacific Community, and he's a wood processing technician, part of the Natural Resources Management team at SPC's Land Resources Division. And Ali Ferretti Bola's presentation today will explore the forestry responses to CRB in Fiji, linking pest delimitation, senile coconut management, so that's the management of dead coconut palms, and engineered wood products. Bola, the floor is now yours. We can see your presentation already on the screen, Bola, just that it will need to go to slideshow before you start. I see that you tried to click the slideshow button to get that underway, but we're still seeing well now, right now we see the slideshow, but you might want to go to display settings and change that a little bit, because we see the presenter notes as well. That might be on the display settings. That's just above the slide. All right. Good to go, Bola. Thank you so much.
Sorry about return. Thank you a lot for the patient, guys, team. Aloha, my name is Alivere Timbolangazi. I'm the wood processing technician under the Natural Resource Management Program, Land Resource Division of SPC, based here in Suva, Fiji. Today I'll be presenting on how Fiji's forestry sector is responding to the coconut rhinoceros beetle, how we integrate this work with the engineered wood product project. This presentation links two important areas: biosecurity and forestry innovation, and show how serious pest problems can become a sustainable opportunity through smart management and research collaboration. The EWP project, supported by ACR, focused on turning senile or CRB-affected coconut palms into high-value engineered wood product while reducing pest habitats. Over the next couple of minutes, we'll explore how these efforts connect, from field monitoring to pest limitation to wood processing and market potentials. Before we begin, I'd like to acknowledge and thank the organizers of this symposium, as well as participants from region and national agency who make this kind of knowledge share possible. The Engineer Wood Product Project is a flagship initiative by the SPC Land Resource Division under the Sustainable Forest Land Management Program, Pillar 2. The goal is to sustainably utilize senile coconut palm in Fiji and across the Pacific. These palms are often low-yielding or affected by the coconut rhinoceros beetle, yet their wood contains significant value when processed appropriately. The project demonstrates how biosecurity and forestry innovation can be combined to generate economic, environmental, and social benefits. Over the next slides, we will explore the entire EWP value chain, including harvesting, grading, processing, and market delivery. The project also provides training and capacity building for local communities, helping them turn what was once a low value or pest problem into a high value resource. Senile coconut palms, generally over 60 years old, are traditionally seen as low value because they produce fewer coconuts. Many of these palms are also infested by CRBs, making them a pest risk. However, the wood from these trees is dense, durable, and suitable for engineered wood product. By harvesting and processing these trees under controlled conditions, the project reduced CRB breeding grounds while recovering valuable timbers. This approach transforms challenges into opportunities, creating economic value while supporting biosecurity and sustainable forestry practices. Senile palm are often low-yielding and heavily affected by CRB, which make them a risk for ongoing infestation if left unmanaged. Harvesting these palms not only reduce pest habitats, but also provide a new resource for engineered wood product. The approach linked pest management directly into economic development, showing that forestry operations can address both environmental and livelihood challenges simultaneously. The CRB management is integrated at this stage, ensuring that the harvested palms are safe for processing. The engineered wood product value chain demonstrates how senile coconut palms are transformed into high-value timbers produced while minimizing CRB risks. Each stages integrate biosecurity, forestry practices, and industrial innovation. The selection of senile palm trees, the processing begins with field identification of senile or low-yielding coconut palms suitable for harvesting. Forestry teams inspect each palm for trunk straightness, soundness, and external signs of CRB infestation, boring holes, frost, and crown damage. Trees showing advanced decay or severe infestation are excluded. This early assessment ensures that only structural sound recoverable logs enter the production chain and that infested materials can be treated or destroyed on site. Harvesting, trained operated fells selected palms safely, removing crowns and frown immediately to prevent beetle spreading. Debris and soft materials are either chipped or burned into controlled manner following Ministry of Forestry protocols. Logs are cut into standard size and transported promptly to processing sites. This minimizes field exposure and disrupts CRB life cycles by removing their potential breeding sites. Grading. The process yard logging are individually inspected by grading for both quality and pest risk. Key grading criteria including diameter, straightness, internal condition, and visibility of CRB damage. Logs are marked as low, medium, or high risks, determining how they are handled downstream. Accurate grading ensured sufficient use of raw material, consistent veneer recovery, and compliance with CRB management standards. Before peeling, senile coconut logs undergo a steaming process, also known as conditioning. Logs are placed in the heated chamber and steamed pit for several hours. The purpose of steaming is threefold. It softens the dense coconut wood fibers, making peel smoother and reduce veneer breakage. It stabilizes moisture content and improves the uniformity of veneer thickness. It serves as a biosecurity function, as a heat treatment helps destroy any residue CRB larvae eggs within the log. After steaming, the log are directly moved into the peeling machine. Conditioned logs are peeled onto a rotary leather to produce continuous veneer sheets. Operators carefully remove any other bugs and damaged section before processing. Veneers are trimmed and sorted according to the thickness and surface quality. Defective or infested materials is discarded and destroyed under supervision. This stage transforms logs into clean, uniform veneers suitable for engineered wood productions. Veneer are dried in kilns or solar dryer to a moisture content of around 6 to 12%, depending on the product type. Drying veneer are then pounded using phenolic or melamine formaldehyde adhesive or pressed under high temperature to form laminated veneer lumber or plywood or panels. These products are tested for strength, stability, and durability compared to traditional hardwood. At this stage, all CRB biosecurity risks are fully neutralized. The finishing EWP products are cut, graded, and labeled for sale. Each batch includes documentation, verifies sustainable harvested, and CRB safe processing. Products are supplied to local furniture makers, construction firms, and export buyers. Residue materials such as offcuts and sawdust are repurposed for energy, compost, or cheap wood, ensuring zero waste and a circular economy model. The value chain represents an integrated approach where each process steps double as a biosecurity measure by linking pest management, sanitation, and industrial production. The EWP project turns senile and CRB affected palm into valuable economic assets while protecting Fiji's forest and agriculture landscape. Research under EWP project focused on ensuring that coconut wood from senile palms can meet rigorous industrial standards and perform reliable in commercial application. Comprehensive mechanical testing have been conducted on laminated veneer lumbers, plywood, evaluating characteristics such as strength, durability, bend, and shear resistance. These tests have shown that properly processing coconut wood can perform comparably to many tropical hardwood, demonstrating it's suitable for furniture, construction, and other engineered wood application. This research not only validates the commercial potential of coconut wood, but also reinforces the value of integrating pest management into the harvesting process. By carefully removing senile palms that are potential breeding sites for coconut rhinoceros beetle, the project addressed biosecurity concern while stimulating creating high-quality timber products. The finding highlights that innovating process and research can transform what was once considered a low value or pest-prone wood into a commercial viable resource. This approach illustrates how science, forest, and industrial practices can work together to create sustainable solutions that benefit both environmental and local communities. The social economic impact. The project has trained local workers, many from rural coconut growing areas, in modern harvesting, grading, veneer production skills. This capacity building empowers them to participate in a value chain that adds income where previous senile palms were seen as waste. Communities gain long-term employment and local operators benefit from new technology and safely protocols introduced by SPC and the Ministry of Forestry. Partnership with industry players such as Long Investment Limited ensure continuity of skills application and incoming generation even beyond the project's ending in January 2026. The EWP process supports Fiji's sustainable forest management goals by converting old or CRB-infested palms into usable veneer. The project prevents open field burning and reduces carbon emissions. Removing senile palms also disrupts CRB breeding sites, helps contain pest population across coconut growing regions. The entire production chain embodies circular economic principles in ensuring that all materials from veneer to offcuts is reused or recycled. This integrated system created environmental and economical sustainability side by side. Together, these two pillars, people and planet, demonstrate how the EWP model can be scaling across the Pacific, linking pest control, livelihood security, and green industrial development. The EWP project has faced several operational challenges. Remote plantation make transport and logistic difficult, and the peeling line at Long Investment Limited has caused temporary delay in processing. Despite these challenges, the project has provided key lessons integrating CRB biosecurity into each stage of the value chain, prevent pest spreading while maintaining log quality. Community engagement is vital, and farmers and contractors are empowered to transform senile palm into an economic product. Finally, as the project wraps in 2026 in January, the Ministry of Forestry have continued oversight, ensuring the successful delivery of the finished product and providing a model for other countries in the region. Oops. Just give me a minute here. This is kind of.
No worries, Aliveretti. Did you want to go back a couple of slides?
Yes.
All right. And while you are doing that, for those of our participants that are still online, if you do have a question for Aliveretti, we do have some time before we wrap up this presentation and move to the last one for the day. In fact, the last one for the symposium. If you are thinking of asking any questions, feel free to pop them in the Q&A. All right, Paula, back to you, bro.
You guys can still see my presentation, or?
Yes, we can, but you might need to switch the display mode once again, like how we did the last time. Excellent, thank you.
Thanks for that, bro. Okay. Well, the CRBG is the most invasive biotype in Fiji, causing crown damage, reduced crop copra and coconut heel, and sometimes killing palms. Larval development in decayed trunks, crowns, and organic debris, making senile coconut plantation a breeding hotspot. Forestry teams actively remove infested palms and sanitize the area to reduce pest population. After clearing, sites are prepared to replanting so new palms can grow in a safe, low-risk conditions or other crops. Information from field surveys and monitoring guides safe harvesting under the EWP projects and ensuring biosecurity and sustainable use of senile palms. Without proper intervention, beetles would continue to spread across the plantations. Teams monitoring high-risk areas, including senile coconut stand, using surveys, mapping, and visual inspections. The removing of infested palms and debris reduce the breeding habitats, directly controlling CRB populations. The logs are sanitized before transport to prevent larvae and eggs from entering processing lines for the EWP projects. The strategies scheduling and doing ensures harvesting occur in a low-risk windows, minimizing beetle spreading. With the community engagement is essential. The local farmers and contractors participate in CRB's sanitation, monitoring, and awareness reinforcement project impact. With the Ministry of Forestry have taken over the oversight to ensure that all operations follow biosecurity protocols, combining pest controls with sustainable timber utilization. Just to add, although replanting coconut trees is not directly part of the EWP project, many of our coconut landowners around South Savu and Riketi have started replanting coconuts in harvesting areas on their own agenda. The SPC, in partnership with the Fiji Ministry of Forestry and stakeholders, conducted monthly delimiting surveys across 5 key sites in Fonolevu, Fiji, the second biggest island in Fiji, 1 in South Savu with 20 traps, and 4 in Riketi, totaling 22 traps. Monitoring occurring 4 times per month, providing temporary trends for CRB activities. Population trends, with the beetles numbers increasing during the second and third monitoring, indicating peak activity period. By the 4th monitoring, there was a significant population decline. Laboratory test confirms that the presence of a virus infecting the beetles, which likely contributed to the drop in numbers. These results allows forestry teams to target interventions, prioritize safe harvesting areas for EWP logs, and integrate biosecurity measure into field operations. Survey outcomes are also informed community awareness and training, highlighting areas where CRB pressure is high or where sanitation is most critical. By converting senile coconut palms into engineered wood product, the EWP project provide a sustainable tools for CRB control. This approach links biosecurity, economic development, and sustainability, creating a model for the Pacific region. From the team here at SPC, Land Resource Division, Forestry Management, that's my presentation for the day.
Thank you so much, Aliveretti, for those updates and for an interesting twist on CRB management around the use of senile palms for processing into what we call engineered wood products, or cocoa veneer. We do have a couple of questions in the Q&A, so I see that we have a minute or two before we move to the next presentation, so I'll quickly ask those. Aliveretti, for your response. The first one from William Pereira, and he's asking, do termites damage coconut timber products? Are there any observations around that?
Thank you, William. At the moment, we have not actually harvested senile coconut trees. We had trials with young trees as 40 years old. We haven't seen any termite damages on the trees that we have logged so far, but we are definitely keeping an eye on that and keeping a record as well.
And that would also mean no damage observed on the products or the veneer produced from those coconut palms?
So on the product end, we have not seen any termite damage, but definitely we are keeping on track on any logs that's coming in and out, and also on the product end as well.
Thank you for that. And perhaps very quickly, just taking another 15 or 20 seconds to this. Question from Dr. Trevor Jackson. Do you have a plan for controlling CRB in breeding sites around processing areas, that is sawmills?
Thank you, Trevor. Not at this stage, but I think we will take that to note. That is something that we will put on as a guideline as well, to make sure that we have control in our sawmill. We just didn't realize that we bring the logs straight into the to our steam chambers, and that's pretty much it. But thanks for the pointers.
And very quickly, the last one, Aliveretti, this is just perhaps food for thought for now. Could the logs be steamed in the field? This could potentially reduce CRB from being moved with the logs. Any thoughts around that?
Definitely, that can also occur on field. It's just a matter of bringing on the materials and the operating cost of doing that. But definitely, most of the areas here in Vanolevu, the plantation is closer to the steam areas, so it is just around a 10-kilometer area.
All right. Thank you so much, Aliveretti, for the presentation. And I note the conversations or the discussions in the chat as well, and the confirmation around the CRB variant that's currently recorded from Fiji. So thank you, Dr. Mark Arrow, for that. I'll hand you over to Dr. Mike Melza from the University of Hawaii to take us away with the a wrap-up of this year's symposium. Mike, over to you. Yep, you can see the slides, and you're on slideshow now.
All right. Well, first of all, yes, thank you for sticking with us for these 3 days. We are finished, but we're not quite done yet. First of all, the organizing committee would really like to thank the audience for joining us over these last 3 days. It's people like you that are interested in this subject and that have a passion in the battle against this invasive pest. We really appreciate it. And it shows how, if there's a silver lining to CRB, it shows how we can sort of unite a region in terms of, a passion for this battle. If it wasn't for CRB, there's so many on this call, whether they're presenters or audience members, that, we wouldn't have had the opportunity to meet. With that said, I'd also like to, even though this slide says thank you from the committee, I'd also really like to thank the committee. Mahalo nui loa to Keith, Franny, and her team here in Hawaii, and especially at Elliot who really spearheaded this work and really got things organized. And then Vinaka Vakalevu to both Ritten and Mark, our friends and collaborators down at SPC, for their work in getting a lot of the folks in the South Pacific involved in this. And so, I was really happy to see such a regional approach to this and hearing the voices from all over the Pacific. And then finally, thank you to our presenters for sharing your knowledge and your willingness to share your knowledge and experiences throughout the Pacific. And we hope you work hard over the next year so that you can join us in person in 2026 and give us updates. We really want to hear your voices, but we also want to hear the voices that we didn't hear in this. And so that's where that audience participation in terms of suggestions for symposia for next year and sessions is going to be critical. So, you know, please keep in touch and keep an eye open for this symposium that's going to happen in 2026. And then I believe there's going to be a post webinar poll that's going to be coming out. I don't know if I can there it is. Look at that. So feel free to fill this out. Hopefully there's enough of you still around, and we'll give you some time to fill this out. I don't know if anyone else on the committee has more poignant things to say than I do. I'm definitely not the person to have, you know, smart things to say at the end of this. So I'll turn it over if anybody else wants to chime in.
You did great, Mike. I think you covered it.
Okay, I don't know if there's any other slides after this one, but I guess we want to keep this open just so that people can poll. So with that said, again, thank you, everybody. And we really look forward to hearing your voices in person next year and seeing those faces.
Yeah, thanks everyone for attending, and we hope to see you again next year.